att se så många här idag. Vi börjar med att göra det jag gör nu, nämligen att sätta den här på ljudlös eller flight mode, så vi inte stör några talare eller livesändningen. Betyder det där att vi kör nu eller? So, it's my great pleasure to welcome you all to this year's first BioStock live event here in Stockholm. We have a pretty extensive program today. Actually, we had 10 companies who were going to present today. Unfortunately, there's only 9 left because of not the coronavirus, thankfully, but some kind of bug. So 9 companies will be presenting to you all today. Sorry. And as far as the companies go, it's a wide selection of different areas they are active in. So it's going to be a very interesting day, I'm sure. First out to present today is ISR, or Immune System Regulation, and will be presented by CEO Ola Winqvist. So, welcome, Ola.
Thank you. Well done. Good morning. ISR is a spin-off company from Karolinska. We're a life science company in phase 2 clinical trials, and we are about to celebrate our 3rd year being listed at First North Nasdaq. ISR is not focusing as many other drug companies, where they are focusing on the virus, the bacteria, or the tumor. ISR is focusing on the host. We are developing a set of drugs that will stimulate the immune system. The immune system will then target the virus, the bacteria, the cancer. Our first goal is to work with HIV. HIV, as you know, is a huge problem worldwide, where more than 37 million people are infected. 35 million people have died so far, and about 1 million people die every year. Although we have wonderful drugs that, if you take them properly, will give you a nice, high quality of life, you can never be cured. Our entry points is with an understanding from cats suffering from FIV, the cat equivalent. Colleagues of mine castrated cats on their street on an epidemic, and they survived. They cleared the virus. And years of research has demonstrated that it is a compound GnRH, gonadotropin releasing hormone, that is produced and causes effects that cures the cats. So when we stimulate the immune system with GnRH, T cells that carry the virus, this HIV virus, will be recognized, killed, and eliminated. It is like raising a plague flag. So an immune cell that is infected with the HIV virus, when stimulated with GnRH, it will start to upregulate flagpoles, class 1 molecules, carrying the HIV flag, which can then be recognized by killing cells and thus eliminate the disease. Carrying out a clinical trial in South Africa, we have demonstrated that using GnRH is safe, very well tolerated, and it diminishes the viral load of the HIV virus. Again, it stimulates T cells, and it stimulates specific T cells that recognize the HIV virus. What became very interesting is that after 4 weeks of treatment, we had a follow-up period for a month, and then the effect continued. So viral loads went down and specific T cells increased. That suggests that the method we are using causes a state of vaccination for the patients. And that is in sharp contrast to what you see when you stop ART medication. Within 10 to 12 days, the virus skyrockets again. So this leads us to another question. Can HIV be cured? Yes, there are 2 cases that have been described where you have cure. And that is because the virus has a certain entry point. It uses proteins on the surface of T cells. And 2 patients that needed a stem cell transplantation for other reasons, also having HIV, the stem cell match was chosen that one of these entry points was mutated, inhibiting the entry of the virus. So the virus could no longer enter into the cells. And by wiping out the immune system carrying the virus, they were cured. However, unfortunately, this is something that's going to be available for most patients. That's going to be a small, minute fraction. So what is the challenge with HIV? HIV is carried in our genome. So within our T helper cells, in our immune system, we have a mole. The HIV virus inserts its DNA into our chromosomes. And that means that the disease is lifelong. This is something we are trying to address right now in a clinical trial here in Stockholm and in Gothenburg, ISR 03, where patients will be randomized either to be followed when they are on perfectly well-designed ART regimens and their virus loads cannot really be measured, or they will be treated with GnRH during 3 months and then a 3 months follow-up. The study is going on now where we are recruiting patients, and more than half the patients have already been evaluated. Recruitment, as in many clinical trials, has been a little bit slower than anticipated, but one has to realize that many of the patients are completely well, and their desire to participate, coming to the doctor 8 times, getting injections just for a clinical trial, is less than anticipated. But we are getting there, and we are actually recruiting additional sites to get the study finalized. Another challenge with HIV is ART resistance. Like bacteria, the HIV virus can develop a resistance, so it no longer is responding to the treatment. WHO came out with an alarm report 2018, naming that 10% of the population infected have ART resistant. 1 in 10 being newly infected will actually be infected with a resistant strain. Women that start their first line of ART treatment are twice as likely to have a resistant strain. And what is even more concerning is that children born to women infected with HIV have a very high risk of getting a resistant strain. And that means that you have a very high risk of developing AIDS. It's forgotten almost by a generation that 20, 30 years ago, getting an HIV diagnosis was the equivalent of death. That was a death sentence. So we are now setting up a collaborative program with Berlin. So colleagues in Berlin will conduct a study on ART-resistant patients. A small study on 15 patients, where we during 6 months will treat them with GnRH and follow up how we can eliminate mutated virus and making the patients again susceptible to ART. To remind us about the size of the market, we can see that it's a huge market for HIV drugs, and it's predicted to, in the next 6 years to come, to almost double. And of course, here we believe that we can contribute with an additional set of an immunostimulatory drug that will help in the fight against HIV. The company has an additional line of work where we have our own compound, which has been a modulation of an existing antibacterial drug, azithromycin. By modulating azithromycin, we now have an immunostimulatory compound where all the antimicrobial activities has been eliminated. And this is something we developed for cancer and for hepatitis B. So in our daughter company, Immune System Regulation Oncology AB, it's a spin-out completely owned by the mother company, where immuno-oncology is the highlight. Here we are working together with checkpoint inhibitors to make a new combinatory drug for therapy of solid tumors. The strategy is to build to phase 1, phase 2 clinical trials and then joint forces with big pharma in the field of CPI. Checkpoint inhibitors was awarded the Nobel Prize 2018. And that was because 10 to 20% of patients with solid tumors, such as malignant melanoma, non-small lung cancer, or urinary bladder cancer, responded very well. However, 80 to 90% of the patients do not respond at all. And that is because tumor cells are smart. They need to avoid the immune system. And here is the point where we hope to add to the knowledge of 10 to 20% with a complementary effect, where ISR 50 is an immune stimulatory that would help and enhance the immune recognition of the T cells. Carrying out studies in mice in melanoma, which is a very, very difficult tumor form, we can demonstrate that we add to the checkpoint inhibition function. So another 10 to 20% of effect can be seen in mouse models. So for our ISR oncology program, we are now walking into the immuno-oncology field, another field where there is a tremendous increase to be expected the next coming years. The majority of the growth will be in the immuno-oncology field, where the majority is with immunocheckpoints and immune system modulators. And that's exactly the field where we are in. The ISR 50 molecule seems also to be very useful for hepatitis. Hepatitis B is a disease that is even larger than HIV. More than 300 million people are infected, and almost 1 million people dies every year. Next to smoking, HPV is the second most causative known agent to cause cancer, and this is liver cancer. So our ISR 50 has a unique position because we stimulate a particular cell, the Kupffer cell of the liver, which is the immunostimulator of T cells. And by providing ISR 50, we will stimulate the patient's own immune system to be able to recognize infected liver cells and eliminate the B cells. There's a huge unmet need. There are no drugs really available. There are some small signs that you can inhibit and postpone the growth, but there is no cure whatsoever. And we believe we have a position here where we can prevent the development of cancer and treat both acute and chronic hepatitis. Our strategy in ISR for HPV is to move our activities to China. We will focus on the biggest market in the world. In Asia, more than 60-70% of all cases exist. We know that this market will grow, and it's expected to grow with about 4.4% of annual growth rate to reach a market of about 1.5 billion US in the next years to come. Our business model is now to work with and discuss with companies for a license for the People's Republic of China market, or discuss direct investment in our daughter company, or probably and likely a combination of both. So ISR 50 is then our lead IND product, and that will be used in an injection format for oncology, and it will be used in a tablet format for HPV. We have now developed the first batch of our GMP, and the next one, the larger, a couple of kilograms, will be delivered in February. We have made a collaboration with InnoStar, which is a large, very, very active CRO company with lots of connections to the industry in China, for the final end of the toxicology program, where we will be able to, by the end of the summer, have a finalized report that then will be used for an application for the Chinese FDA to conduct clinical trials. And we hope that they will be able to start in the beginning of next year in China. So with this short overview of ISR Holding AB, I've been telling you that we actually have 3 lines of investigations that are ongoing. HIV, where we have a phase 2A clinical trial that is presently on its end stage, where we hope we will have the results during the year. We will initiate a niche indication of art resistance starting during this year in Berlin. And we're developing our IND product that will be ready to be tested in first-in-man studies in HPV in China and in Europe, preferably here in Stockholm for oncology, where urinary bladder cancer will be our first indication. Late-stage urinary bladder cancer, where you have spread disease, where you have an expected life of about 6 additional months. So thank you very much, and I'll be happy to take questions.
Thank you very much, Ola. Interesting presentation. I'm actually going to turn to the audience first of all to see if there's anyone who has any questions here. And if you do, I have a microphone that you will have to use in order to have our online viewers also hear the question. If no one has any questions, I'll go ahead and ask a question then. I think that the status of the HIV study, the timeline there, what can you tell us about that? How you had some trouble with the patient recruitment earlier last year, and what's the status right now?
The challenge is whenever you're going to do a clinical trial that you have a very, very tight description in order to be included. So we have included more than half of the patients, approaching 30 patients. But we also have 19 failures. They were interested to participate, but they didn't fit the criteria. So they need to have been during treatment for more than 3 years. They can never have been low in CD4 cells and so forth. So that is a challenge for us to actually find. I mean, we are blessed in Sweden. There are so few patients that have HIV. And it can only be men, and they need to be accessible for 8 visits. So that is a challenge to actually find. There are a lot of interested people that would like to participate, but when we tell them that during the next 6 months you need to show up 8 times, that's the difficult part. But when you do the first-in-man clinical study for this indication, where we're actually aiming to wipe out all cells carrying HIV virus. It has never been done before. Everyone else is going for the virus and inhibiting the replication. We are trying to kill all the cells by the immune system. So I don't have a good estimate because it's up to the patients. And as a CEO, I cannot even participate in that part according to good clinical practice. So our PIs are proposing for every patient that comes in to participate. But we realize it's taking too long, and we are, of course, eager because more than half of the patients, we have the results, but I cannot see them before everyone is done. So we are as eager as you. So that's why we are also including more sites that can contribute so we can get the final patients in.
Okay. So tight inclusion criteria,
Yes.
that's one obstacle or a challenge. And the fact that many people can live now with the medication that's available, is
Yes.
that also a factor in this?
Yeah, it is a big factor because they feel healthy. So, by coming 8 times to the hospital, you start to feel sick again. I mean, no one wants to meet the doctor, even if they're nice, and get needle pricks. But in a study that is a first-in-man, you need to have a tight regimen because we don't know, and safety is always our concern. So that is the drawback, that there's a lot of interest, but when you need to participate so frequently. So next study, of course, when we know much more, doesn't have to be so frequent. And if we get the results that we hope for, I think it's going to be the opposite, because when you can demonstrate that actually a few patients, we have eliminated the virus, we will have a lineup instead.
Yeah, and of course, clinical proof of concept will be a huge value inflection point for the company as well.
Yes.
And as far as the the financing of these studies. I mean, you're in the HIV study, and then you are aiming to go into hepatitis B
Yes.
study, and as you said, starting then hopefully next year.
Yes.
Financing is always a challenge for companies of your size and so
Yes.
on. And this December, you negotiated a loan that will take you up until June of this year. Is that correct?
Correct.
Yes. And what will happen then?
Yeah, I mean, fortunately, we have a lot of options. We have very, very tight discussions now going on. I mean, not at least to say that some of the compounds we have may actually be useful for the coronavirus. So we have a lot of discussions going on, and I will be happy to share that later when I know more. But we have many options, including, of course, to ask the market again.
All right. We have a question here. Please use this microphone. Welcome.
How will you positively conclude that the HIV infection is eradicated, since these patients will still be on ART treatment?
It's actually very simple. You measure the amount of HIV integrated DNA, which is actually our primary endpoint. So we will measure in extracted CD4 T cells, which are the carriers of the HIV virus, and ask, is there any HIV DNA? And if it's none, there you have it.
Then it's a jackpot.
If you're lucky, but
If you're lucky, yes. But I mean, even a decrease in HIV viral load will be a huge success, because it has never happened before. So we hope that this is going to be the beginning of the end of the HIV virus.
how long will you follow up the patients?
The patients will be followed a total of 6 months, so 3 months on medication and 3 additional months follow-up. So we do not know if 3 months is enough of treatment, but that was what was available from the ethical committee in the discussion with the MPA. So I mean, tuberculosis, another chronic bacterial disease, you treat for 9 or 12 months. So it might be that we get a signal that shows the right direction. Perhaps you need a combination with checkpoints, or you need a longer time for the treatment.
Thank you very much, Ola, for
Thank
your
you.
time. And you're going to be hanging around here for the break as well, so people can grab you in the hallway.
Yes. Thank
Okay.
you. Thank you.
Thank you much.
Thank you.
Warm hand to Ola. So I am going to be a technician today as well, which is also a challenge. But I think I have the hang of it now. There we go. Our next speaker, Stefan Sova of Endovab, or Endovascular Development. Welcome, Stefan.
Thank you.
It's yours.
So I'm CEO of Endovascular Development, and I will tell you a bit about how we make minimal invasive surgery safer. The invention is an anchor. It's like this. You can look at it afterwards. And it's made of memory metal, and at body temperature it regains its predetermined shape. And you place it in the artery where you need it or where it's useful. And the more you pull in this guide wire, the more it sticks to the blood vessel. When you don't need it anymore, you retrieve it, and you don't use it anymore. This is a Swedish medical company from Uppsala. We are improving tools for minimal invasive surgery. We have patent protection in 3 clinical areas, and certification according to ISO 485, and the CE mark of the product class 3. It's the world's first temporary arterial anchor. It's clinically proven and commercially available. And we are now seeking first private funding for growing sales and product development on the next model, and later on for expanding the market and going for the next generation of product line. Normally, traditionally, you do open surgery, but if it's possible, you prefer minimal invasive surgery with lower complication rate and shorter recovery time. And there are essentially 3 ways to get into the body: via an small incision in the skin, and via gastro canal, or through the blood vessels. And this is what this company works with primarily. Endovascular surgery is made under X-ray, and the instruments and implants is inserted through the artery, normally in the groin, but also other places can be available. These are the products on the market: guide wires, catheters, closure device, introducers, filters, stents, balloons, stent grafts. And the success of endovascular surgery is based on the tools available. So 1952, a surgeon from Mora in Sweden introduced this technique, Seldinger, and it's become the most cited article in the medical area. He is a radiologist and wanted to insert contrast media closer to the target. That opened up possibilities for radiologists in primarily to do new things. So here an example of things that sort of evolved from this technique. The balloon angioplasty, where you used to widen the blood vessels, 1974. 86, the stent, also invented by a Swede, Wallsten. And this is another example. It's the pressure wire for measuring the pressure in the heart capillary, invented in Uppsala. And now its next toolbox is the temporary arterial anchoring, introduced by Christer Ljungman, a surgeon in Akademiska and the founder of this company. Now this has grown a lot. They use 100 million guide wires a year, at least 30 million balloons and stents, and the pressure wire is now up to 1 million. And the major players are Medtronic, Abbott, and Boston Scientific, Terumo, and Cook, and other. So, why? The problem with the instrument is lost position. Then you have to restart the procedure, and this means prolonged operation time. The other one is the instrument gets out of control. It will damage the blood vessels and also maybe puncture it, and that is severe complications. So the solution is stable position for hours during the operation with not corrupting the blood flow, because that will damage the organs. The benefits: high risk operation can be converted to acceptable risk level. It enables procedures previously considered impossible. It less radiation for the patient, reduced risk of kidney damage due to contrast chemicals, less radiation exposure to the surgical staff, and this type of operation is the most radio pressure on the staff in the whole healthcare area. You get cost savings, of course, for shorter operations and less intensive care, reduced risk overall for severe complications and death. Now, the expansion opportunity for Endovab is that patients previously considered not treatable can be operated. And there's also opportunity to transfer operations from specialist clinics, like university clinics, to county hospitals. There are anchoring product, they are anchoring in the blood vessels and fixating an instrument. The first product is the guide wire fixator. The second one is the filter fixator. And I will come back to what it's used for soon. Essentially, it's the same component that makes it works, but you can do different things with the you can anchor different things. And the third one with the patent is the catheter fixator. So one area is the abdominal aortic aneurysm. 6% of all male over 65 suffers from this. So in the Western world, they screen this with ultrasound. And if they find a growing aorta is 2 centimeter in diameter. If it's going up to 5.5, it's close to rupture, and you have to do something. And when the aneurysm reach all the way up to the kidney artery, it's called complicated, and they have to be carried out in a university hospital. It's only 10% of them. The other is considered not treatable or done with open surgery. So the guide wire fixator enables county hospitals to treat these complicated patients. The next area is called embolic protection. When you have calcification of your artery in the legs, it's a large disease. 40% of all people over 70 years of age suffer from this. And when it gets too far, you cannot walk, or you have pain in legs and black toes, you need to do a revascularization, so open the artery. And that is done with balloons, stents, or a small drill, actually. Whatever you do, you cause debris to flow downstream, and that can be blocking artery further down or in the capillary. That's called embolism. That will lead to amputation, may lead to amputation and death. So this filter fixator is focusing this procedure, placing a filter downstream over the operation, and collects all the debris. After the operation is taken away, and you can see how much you saved from the body. The third area is called precision drug delivery. You anchor a catheter close to the target tissue, and then you infuse the drug during treatment period, which can be weeks, and the patient can be ambulant at home. This substance can reach its target without first being metabolized, and with very toxic chemicals, we can avoid systemic effects. For example, in chemotherapy of 40 million of tumor cancers in the world. So the fixator focus on that market. Of course, this company need partnering with other companies, drug companies and other, of course, later on. So summary, market drivers. The minimal invasive surgery is sort of a trend. If it's possible to go for minimally invasive surgery, they will do that. With enabling tools, more patients can be treated, and there are a lot of cost savings in that. Endovab, as the first enabling product in this area, temporary arterial anchoring. Device is a single-use tool. It's not an implant, and there's no active components, which means it's easier regulatory-wise. It's one technology, and there are several clinical fields that can be addressed. First product is now clinical proven and commercially available. And the next model, the filter fixator, is under development. There are already prototypes. Thank you.
Thank you very much, Stefan. You're super quick.
Sorry.
No, it's perfect. Obviously, I have a question from the audience. Here you go.
Just a quick one. A comprehensive, not too detailed explanation how this anchoring goes about. How do you do it?
Would you like to show on me?
I lost the part. The anchoring is introduced to the vessel from
No, wasn't there.
the first animation you saw. You pass it into the artery through the artery, and you end up in the, for example, the renal artery, and then you release it in that place, and it will regain its predetermined shape. It's a memory metal, and will then stick to the wall. And the more you pull it, the more it will stick, and then you can remove it. So using this guide wire fixator is like placing the railroads into the system. You have, for example, 3 or 4 of those going into different peripheral artery, like the arm or the renal or the liver. And then you end up with 3 or 4 of these wires outside the body, and you can then arrange the implants outside the body, and then just push it in and release the implant if it's a stent graft or if it's a heart valve or something. So this is not a cure or a treatment itself. It's just helping the doctors to do. They can go closer to sensitive areas like the brain or the heart, which they don't dare to do right now. But I don't know if this answer to your question. Okay.
Find it interesting then. Like you said, it's all about the tools that the surgeons can have available to use. That's how safe it can be and so on. What are the biggest challenges in introducing a new tool in this particular area of activities, endovascular surgery?
Yeah, we are communicating with 5 clinics in Europe. They are leading clinics in this area. And so they've been part of this safety study, which is published. And they will now try it out in different applications. This aorta thing is quite complicated compared to other things they can do. And they are now, they will publish their results. This is very helpful in this area, on this area. And that will be collected, that information, and then we will start more dedicated studies for using it in certain cases. Was that an answer?
Sure. And in terms of market entry and market uptake, what will be the sort of the gatekeeper, or what will actually make this take off big?
Well, once you have to publish thing.
Yeah.
And it doesn't take so much since it's scientifically very interesting. This is the first line of products in this area. So that will be published scientifically and presented on conferences. That's the starting point. Then the clinics has to find way to use it and be trained by us or key surgeons in the hospital. And we don't think it will be used in every case. It's just we need to go through this very complicated vessel system and to be sure. And that's on the shelf. You have to have all sizes available when you need it. So they will now they have ordered products to have in their shelves. And when the interesting case comes, they will use it. We follow that up, yeah.
And when you say they have ordered it, how many sites or clinics are we talking about, and what's the prognosis going forward for that?
Okay,
expansion?
the 5 key clinics in one in Holland, Germany, Switzerland, and Uppsala, and there are 3 of these county hospitals, Falun, Gävle, Eskilstuna, also ordered this to have in stock. But the forecast for this year is fairly conservative. It's only 56 patients we are going to sort of focus.
Okay.
And then when we publish the result, we can use that in our marketing message.
Yeah. We have one more question.
Could your anchoring device be added to the already established technology, like from those competitors you mentioned, Medtronic and so on? Or do you need to buy the whole equipment from you?
No, very good question. We have been discussing with Medtronic, Gore, Cook, to combine this with their stent graft, for example. And they're very happy technically, but they say we need to see clinical evidence, then we can start to combine it with our products. So we are a tool for every purpose. And we also hope that the next generation of products will, you can find other things to fixate. And you can also go into other canals in the body, not only the artery. So there are opportunities here. And they all support our sort of endeavor that to anchor things. For example, down in the leg, to go down to this artery to revasculate that, you have to go in from the other side, over the aorta, down to this side, and do the whole thing. And if things slide around, you damage the blood vessels, the epithelial, so it will be you have more problems than before. So anchoring things, everybody thinks that of course we should have that. If you don't have anchoring inside, you have to do the anchoring outside. So the staff will keep track of all things on the table that's going into the body to make sure they don't go too far or fall out. So that's what they're doing today.
We are almost running out of time, but I was like, since Endovab is still a private company, are there any plans of going public?
Yeah.
Okay.
In the future.
That was one short question, one short answer. So what about the business model? What are you aiming for? Are you going to reach the market and sell all over the world by yourselves, or are you going with the normal way to find a partner?
The strategy is to show in key countries that we, together with a very competent distributor, it doesn't have to be big, but it has to be competent, and show that we, within a certain number of years, can reach a certain number of part of the patient available. So, for example, in the aortic side, it's only 300 patients in Sweden that's sort of eligible. So, but showing we can take that part, that could be multiplied for all the world. That's what we then hope, that the big companies will have a distribution deal with us. And then,
1 minute left. Isn't there a big risk chance of them just snatching you up if they see that this works?
It's always a price. Yeah. I think like this radiomedical system I mentioned here, that was acquired by St. Jude 2008. We were 350 people. I worked there. 350 people, and it was $200 million sale. And now that product is now selling for 4 billion kronor. So, but
Say no more.
say no more. but we have to show clinically stability, commercially available, commercial use in the field before they really react.
Yeah. It's been a pleasure listening to you, Stefan, and we look forward to following this interesting journey. Thank you very much for today.
Thank you.
Thank you. So make sure that if we find your device, we will pick it up before they vacuum clean the place. And here we go. So our next speaker is Anders Hägerstrand, CEO of Annexin Pharmaceuticals. Welcome, Anders.
Thank you.
Leave this.
Thank you. So it's a pleasure to be here. See some familiar faces in the audience. So my name is Anders Hägerstrand, CEO of Annexin Pharmaceuticals since October of last year. I will start talking about the fact that we are actually in the midst of a share issue. We're raising 61 million Swedish kronor at a subscription price of 1.15 Swedish kronor, which will mean, if you multiply it by the current number of shares, not beyond the share issue, but right now it's the market cap is only about 20 million Swedish kronor. We're in the midst of the subscription period, and you can find the instructions and the prospect on our website, Red Eye's website, and also on the Finansinspektionen. The major shareholders are all contributing. 50% of the share issue is already covered by them. Another 50% is covered by guarantors, external, and some other also existing shareholders. And the founders are also involved in contributing to the share issue. So, about the company, established in Stockholm back in 2014, it's a spin-out from Karolinska Institute, where so the scientists and the founders still involved in the company found that Annexin A5 is a protein that can not only be used in research to identify dying and damaged cells, it can also be used to treat patients potentially. They started off with a fairly rare disease and proved that in this disease, the vessels in these patients were full of Annexin 5 for a reason, and they found out that this could be potentially a treatment of this disease. So they patented the use of Annexin 5 to treat various diseases. That's really the opportunity we have in the biologics drug field, where you have a use patent, and you can also cover it by production patents. So they started to do that, and we're now a small company based in Stockholm. We're 4 full-time employees, and we have a number of consultants for specific questions and fields. We are world leading in the field of Annexin A5, and we're the first company to be able to manufacture the drug large scale GMP for even with quality suitable for the market. Focusing on vascular disease, I'll come back to that. And again, our patent portfolio is about the production of Annexin A5 as well as covering the use of Annexin A5 to treat many diseases. There are almost too many opportunities for this drug. It's a really old, we call it ancient protein. It's in all of us here. It's been sitting on the inside of every cell. It's very, very present in the vasculature. So on the inside of the vasculature, including the heart valves, the aorta, the arteries, the veins, the capillaries, everywhere. There's a cell type called endothelial cells covering the inside, and they're really rich in Annexin A5 in the membranes. What Annexin A5 does is when cells get injured, it sort of pops out on the outside and scavenges and binds to certain fatty acids, which are known to be inflammatory and dangerous to the body. So it pops out, binds them, and neutralizes them. Then the whole complex is excreted in the urine. So actually the drug is called, or the protein is called a protective shield, band-aid, missile, bloodhound, etc. So it's really known for that function. What our founders did was to sort of turn this into a therapeutic opportunity. So again, many opportunities for this molecule. We call our molecule ANXV, which is our version of Annexin A5, which is and very, very, very similar to the endogenous natural protein. We're looking at some different kind of diseases. We're looking at smaller indications like retinal vein occlusion, which I will describe in some detail, sickle cell disease. And if we jump to Ebola over here, and even speculations about being possible to use it in coronavirus disease, these are sort of where cells are getting injured in the vasculature. These are blood cells getting clotting. They are sticking to each other. They cause disease. There's a crisis of release of the various inflammatory compounds in Ebola, which can possibly be treated by Annexin A5. Whereas in vascular disease, in the heart and in the vessels, we can really protect also the vessel wall, not the cells inside the vessels, but in the vessel wall. Apart from that, there are also sort of a basic use of Annexin in basic research is used actually as a molecular imaging tool. So before we started to work with it, it was used to find damaged cells and damaged areas in the body by tagging a small radioactive compound to Annexin A5, injecting it. And since this radioactive decoy is possible to detect with the camera, you can detect where in the body you have a sort of a deceased cells or dying cells. So we've already been in the clinic, but as a diagnostic tool or as an imaging tool. You can also tag an anti-cancer drug to Annexin A5 and inject it, and then the Annexin 5 having its missile or bloodhound capabilities, it will find the tumor, find the dangerous site, and deliver the anti-cancer drug specifically to the tumor, not to the rest of the body. So it's a very smart molecule. Some other companies are considering sepsis, which is blodförgiftning in Swedish, also scarring, other versions of oncology, as well as it's possible to use it obviously also in animal health. So when you work with biologics, you have a challenge as a small company. You have to produce and spend a lot of money and time and effort to produce a drug. Tens of millions of Swedish kronor has to be spent on making it your compound that you can put forward into preclinical and clinical trials. So the company has done that for a number of years. We are very proud and happy about the technologies what we have. It's an efficient, cost-effective, high yield, and patented procedure. So we make this in bacteria. We get the source molecule, which we call the drug substance. We dilute it into a solution. It's really an intravenous solution intended in a small vial, which will then get to the clinic. First, of course, obviously, you have to have the drug produced, and you have to test it in animal studies, which we have done. This is according to guidelines, but some of you that may have followed the company would know that we had struggles with the findings in the rat. So we have to go think about what really happened. We have to explain why this reaction occurred. We went to the Swedish Medical Agency and asked them how should we deal with this. We got some advice. We went back to the drawing board, made a lot of new studies that were ending in the fall of last year. Now we are ready. We understand why the rats react in the way they do, and we also have another species which doesn't react at all, which is closer to humans. So we're pretty comfortable now that this reaction in the rat is what it is. It's important to follow and understand. But we're comfortable that we can actually follow any such risk in first-in-man studies that are to follow. Clinical development, we will apply for first-in-man trial shortly. We have announced that we do it early 2020. And you start with one single dose, intravenous then, for 1 day. Then you go dose by dose upwards. Then once those single administrations are done, you go with multiple administrations, because we feel that Annexin A5 treatment or NXV treatment will be for a maximum of 5 days. Intravenous infusion one day, one infusion every day, and that will cover all, in quote unquote, indications that we think are important, because we are wanting to treat this the acute vascular crisis in a patient with an ongoing clotting of cells or with an injury caused to the vascular system. That's where we think the Annexin A5 can make most sense. Clinical selections, and among all these opportunities, retinal vein occlusion, it's a disease that you probably haven't heard about. It's still very problematic to the patients. It's a sudden blindness occurring. So the perspective is almost literally what you see on the left there. The patients go blind, more or less fully blind, all of a sudden, without necessarily any other sort of explanation to it. They get the diagnosis, And the cause of this disease is really a blockage of a vein in the retina. Retina being the area of the eye where the photons are really hitting the body and translated into images. But this area is where all the light is translated into the image eventually. So if you don't have a healthy retina, you cannot see. And if you have the bloodstream with arteries to the capillaries feeding the eye with blood or the retina with blood, and you have a blockage in the vein that would take the blood back to the heart eventually, the blockage of the vein will cause a sort of a stop in the flow. You will get a back stop, and you have edema and swelling, and then the cells will die in the retina, so you turn blind in the end. So why we focus on this one, I'll come back with some more details. It is the second most common cause of blindness. If you don't get the treatment, you have a 10% chance of getting any improvement after you have the diagnosis. With standard of care, which is injecting drugs straight into the eye, you have about 30% to get a meaningful improvement. And there's no current treatment that really addresses the forming of the clot in the vein. Current treatments are really there to stop the swelling and stop the pathological inflammatory process downstream of the initial clot. And we know that the red blood cells from these patients, when you put them in vitro or out in the lab, you can see they clot to each other for some reason which is pretty much unknown. And importantly, I'll come back to the mechanism later, but it's really 28 million people in the world living today that have had RVO at some point in life. Some get a second event as well. Standard of care is very expensive drugs, again, injected into the eye. It can be done one single time every 6 weeks or so, or every month during the course of half a year, that have some improvement of the risk of blindness. It's a huge market. For us, it's definitely enough. It's a $20 billion market. And drivers are, of course, the fact that the people get older and have more atherosclerosis and more diabetes. And we have shown, or others have shown, that if you take these patients' blood cells, as I mentioned, they clot together, but if you put Annexin A5 into the mix, they will not clot anymore in the same fashion. 70% reduction of the actual stickiness. And if you put them together with the vascular cell I spoke about, endothelial, they will not attach to the endothelial either. So we think that by injecting intravenously, Annexin will reach the eye and then potentially even make the clot go away or stop the clot from growing. How we want to move forward with this interesting idea. So the idea would be that we use the current or the expected amount of money that we get from the current share issue to do imaging, phase 1 study, I mentioned that, but also an imaging study in RVO patients. So when patients get into the clinic, get the diagnosis, we will collaborate with specialized teams, with ophthalmologists as well as the imaging specialist. They will have Annexin available. They will attach the radioactive molecule. They will fuse it into the patient. And we can follow then the binding of Annexin A5 in the eye by certain cameras. So we can prove the point that our drug reaches the eye and in the right place in the veins. Next phase, which will be beyond the currently ongoing financing, is to do proof of concept studies. And the eyes, we're blessed with addressing the eye, because in the eye you can actually do a lot of imaging. It's the mirror of the soul. It's also possible to actually look into the eye via the lens. So we can see inside here is the retina and the blood vessels of the retina. To the very right here, you can see a blood vessel. You can actually follow the red blood cells in the vessels of the eye. So the hypothesis here is that we will be able to look into the eye. We'll be able to see the blood vessels. We will see the blood cells actually being affected by Annexin A5. And I could not have had a better pre-speaker than the one I had. So we cannot resist the cardiovascular field either. So it's really when surgeons go in and do things in the veins or in the arteries with balloons and what they do. They do good, obviously, but they also injure the vessels. And this injury of the vessels, putting stents into the vessel, mean that they, of course, open the vessel, increase blood flow, reduce the risk of having the pain in the leg caused by the atherosclerosis or pain in the heart caused by the atherosclerosis. But actually, the stents, there's also quite a lot of problems with them because the narrowing reoccurs, shown down to the right here, where the stent is there. This is the cross section, and after 6 months, it may look like this. So it's already narrow again. So it is a problem, especially in some patients. This is a very common phenomenon. So given what we know about Annexin, we know that it binds injured cells. So when the surgeons go in to do the catheterization, they will destroy a lot of tissue. This is an illustration of an animal where someone has done, to the left you see nothing almost, to the right you see an injured vessel where we have injected fluorescently labeled Annexin. You see it binds to the injured vessel, and it will then scavenge and protect the cells from the inflammatory process that will come afterwards. Patients with problems in the leg, in the arteries of the leg, to the right here, one patient have been injected with Annexin A5, and you can see it binds to the actual position where the dilatation or the balloon catheterization was done. So, hypothesis here that we will be able to, in the long run, protect the vessels after any kind of vascular surgery being done. Could be heart surgery, could be this kind of vascular surgery. We want to prove the point as a start in this big field. So hypothesis here is that we will know that if you do this kind of procedures, you will have a rise in certain inflammatory markers in plasma. You can take a plasma sample, you will see a pattern like this, going from day 0, 1, 5, 15, it goes up and then it goes down again. And we think that if we have the effect that we think with Annexin A5, we will be able to cut this inflammatory effect, which will in turn will reduce the risk of getting back the stenosis or the narrowing. So market overview for RVO, it's a 400,000 patient market, at least for us, which means we're in the $1 million range of sales per annum. We intend to be possibly taking this drug to all through the market ourselves, while for the cardiovascular indications, we think we need to collaborate with larger pharmas. Regarding licensing deals, there was one Annexin deal I'd like to point to, is the one in phase 1 Annexin A1, another Annexin, was licensed by Bristol Myers Squibb for 410 million euros. And also in the ophthalmology field, it's a small field, but still very many deals are done in that field. So we feel comfortable that there are takers out there. A bit of a summary slide, what we intend to do. To the left is where we are right now, what we want to do with the currently ongoing financing or the proceeds of that financing. First in human test or study, imaging in RVO, imaging for another indication, like the cardiovascular indication I mentioned, as well as go to the EMA and FDA and ask them about the development program. You have the luxury of going to them and asking for scientific advice, presenting your case. This is what we want to do, how we want to run the clinical trials. Can you accept that? And the value of having an FDA meeting and the protocol from an FDA, which they have approved the minutes of, it's worth millions, almost literally, for a small company. Not only to be able to describe for investors how to move forward, but also for a licensing partner. They know what the FDA wants. So if beyond that, we want to do proof of concept studies, like smaller concept studies, looking at certain markers, like the blood flow in the eye, like the cytokines in the plasma, and to prove our point before we go to larger phase 2 trials, which are very expensive. Small ones we can do ourselves, potentially. And then, of course, the plan would be to go to final registration for RVO and have multiple licensing partners for the other multiple opportunities for Annexin A5. So in summary, will have a first-of-class kind of medicine. There's no other kind of drug that would do the same thing as Annexin A5 for sure. We're the first one to be able to produce it. We have a patent protecting the production. We're in the sort of midst of filing the FDA, the CTA, the clinical trial application, start the clinical trial this year, be ready by mid next year. We're de-risking the whole development program by imaging and proof of concept studies. And we will also try to grow by potentially partnering some other areas where we're not active ourselves, but we talk to other companies that are using Annexin 5 for other purposes, meaning potential deals. Okay, with that, thank you.
Thank you very much, Anders. So I'll do as usual. I'll turn to the audience first. Any questions? Yeah, one.
I should know this, but I don't. Is there an animal model for RVO?
No.
So how do we then know that this, or not know, but what is the indication that this would work at all?
Sorry. So lost my voice, drinking too fast. So I guess the answer is there is almost never any perfect animal model for any disease at all. So animal models are always surrogate markers for the real model or the real human pathology. So it's really based on the fact that these patients' red blood cells clot in vitro, they clot in vivo in their eyes, and we can inhibit that clotting with Annexin A5. So when we know the binding between the red blood cells that causes the clot is dependent on this specific fatty acid that Annexin binds to and can stop. So it's a theory in that regard. Another model, which is not RVO, but another one, sickle cell disease, is know that this another type of clotting also dependent on the same fatty acid. In vivo, Annexin A5 would stop that clot from forming. Also. So the hypothesis is based on animal binding data, human blood cell data. That's where it comes from.
You mentioned that you're in an ongoing financing procedure, and you're aiming to raise 61 million Swedish kronor. How far will that get you?
It will take us into mid-2021. So the idea with the whole finding is to reach certain specific milestones, meaning the end of phase 1. So now we can move forward into RVO for phase 2, or pretty much any disease we select, as well as having the imaging studies done as well, in RVO as well as in the cardiovascular disease I mentioned. That will be sort of the basis for potential licensing deals, and also maybe to go back to financiers to raise more money for the proof of concept studies I outlined.
So having an asset that you perceive has such a broad potential in many different indications and within the vascular protection field. RVO is sort of you have narrowed it down and you have a clear idea what you want to do there. What will be next? Is it too early to talk about that?
Yeah, I guess so. I mean, we'd like to work with diseases where the blood cells clot for one or another reason. We can sort of stop that clotting, which goes for RVO and sickle cell disease. Ebola is a bit of a different disease in the sense that you will bind the phosphatidylserine, which is the same fatty acid again, that would cause the storm that causes the dramatic development in Ebola. So we think we can reduce that storm. But then, so I mean, that's one area we would like to pursue for sure. And then we start with RVO, probably go beyond that later on. And then in the vascular field, I think we talked about is large indications enough when we have the balloon catheterization. But again, if you think about the fundamentals here, you can protect any vasculature damage to the wall, which surgeons do every day. When they do heart surgery, vascular surgery, I think that's going to be a huge field as well.
Apart from getting the financing done to finance the upcoming studies, then I think you mentioned that there are some small things left to take care of before you can go actually into the clinic.
Well, yes, I mean so what we have done is that we've done the animal studies, which we have concluded. We think we have enough evidence. We have to clean, let's call it clean, the drug from a small contamination that we found in the early fall. We're doing that process right now. Of course, then submitting to the Swedish Health Medical Products Agency is not a predictable thing really. You can end up getting questions that you have difficulty answering. But given the experience that we have in the company, all the consultants, we feel comfortable that we will get through and to be able to start the studies. So that's really the focus that we have right now to get it through to the clinical study.
Would you say that the slight setback in the fall, finding the reactions in the rats, forced you to take another look sort of at the whole package? Is that now a strength going forward?
Yeah, no, it's a fairly specific thing, right? It was actually discovered almost a year ago. Then we had to go back to the drawing board and design the studies and get them done and get them sort of reported. So it's a fairly specific event, which hasn't led us to rethink anything but that. We have the same. The goal has been RVO. I think we've recently expanded a bit, say not only RVO but also cardiovascular disease, in terms of the imaging studies as a first step.
And summing up, what are the particulars of the ongoing finance and as far as subscription dates and so on?
So it's in the midst of it. Subscription last day is 21st February. I think I mentioned most of it. It's guaranteed in various ways, but I hope all of you will try to subscribe, of course. But it's ongoing. It seems to be going well, although we, of course, as a company, we don't really know.
All right.
Another question.
And one more question. Yeah.
The fact that your product is first in class sounds so very nice and interesting, but it's also a major challenge. So what is the major challenge during the next 12 months?
Well, the first in class doesn't really affect you right now. I mean, the challenge is now, of course, that we are sort of moving into the patients with it's not similar to anything but itself. But sort of in that thinking, it has been in man already as a diagnostic tool with the molecular imaging. So at a very low dose, it has been used in humans already with the same kind of binding and bioactivity. So it's just the fact that it's different, slightly different molecule with this molecular tag to it. So first in class is normally a good thing. It's a challenging thing when you come to the FDA. They will want to know a little bit more, maybe more about potential drug interactions, etc., with that which we'll have to work with going forward in the phase 2 program. But I think there's much value to it as well because there's nothing similar to it on the market.
Yeah, we're looking forward to see where you take it.
Thank you.
Thank you very much.
Thank you.
So that actually marks the end of our first session. Now we're going to take a short break, 15 minutes, or should I say maybe 10 minutes, since we ran over a little bit. At 11:20, the next speaker will be on here, and that's AcuCort. So welcome back. So, welcome back after an invigorating coffee break. We let it last a few minutes longer since we have actually one more speaker now, and then there was a company called Lipum that was supposed to be next, and because of illness, they're not here today. So, but we sort of adjust our schedule accordingly. Our next speaker is Anginer, who's CEO of AcuCort. Welcome.
Thank you very much. I'm very happy to be here today to present AcuCort to you. And we are developing convenient oral film relief. And to start with an overview, the company was founded in 2006. It's a public company today, listed on the Spotlight Stock Exchange since 2017. And we also performed a new share issue recently, so we have good capital in the company right now. We are based in Helsingborg in the south of Sweden. And since January 1st, we have a new CEO. I have taken over as interim CEO. And I'm very, very pleased to take the company forward. Looking at activity, we have very significant activity on the main product, AcuCort product candidate. This is an oral dissolving film. It's based on dexamethasone, active substance, and this is all to go into the glucocorticoid treatment arena. And we have great convenience advantages with this oral film. I will tell you a lot more about that. And to say something on the position of the company, this is a first-in-class combination to have this corticoid treatment with an oral film. We are very happy to be well patent protected, and we also have the product trademarked. We had an application made for market registration to Swedish Läkemedelsverket in September 2019, and we have an application for the US market in planning. And to tell you more about the actual concept, ODF is the English abbreviation, oral dissolving film. And it's a cellulose film where we have invented the combination to integrate dexamethasone. And this is a very well-known glucocorticoid. So it's very easy to argue for its safety and its convenience for patients. But the new application with the oral film gives very simple self-administration. And you don't need any healthcare intervention. And this means you can take this medication anywhere and anytime. And that's completely new. We have a patent family protected in 31 countries already. And we have a second patent family granted for the European Union and in application process right now in the US, in Japan, in Brazil, in China, Australia, and Canada. And of course, in pharma, you have to target specific indications. And we have selected these 3 key indications you can see here. The first one is allergy and acute allergy. And I will be going into more detail about this market and the market size. Second, we have croup, which is a pediatric indication. And you can imagine the change if you have a poor baby suffering from croup and the difference to give just an oral film instead of trying to make the baby take 10 pills or something. It's not very easy. And the only alternative to pills today is an oral solution. And then you have the dosing issue, right? How can you tell if a baby has swallowed 2 drops or 10 drops or something? The third indication here is called CINV. Chemically induced nausea and vomiting for chemotherapy patients. And here personally I really see a benefit of the product because here we can help cancer patients who are feeling very sick to reduce the sickness with this very easy administration of an oral film because it's not easy to take pills when you have nausea. So we are working mainly on these 3 indications, but we have also applied for further dexamethasone indications. So in total, we have applied for 8 indications. And the whole point, like I'm already telling you, is the convenience of the product candidate, AcuCort. The package size is the same size as a business card, as thin as a business card as well. And you just open it and take out the film, as you can imagine. So you can bring it with you, you can have it in your bag or in your wallet, to always be prepared. If you have allergy, and you can often be afraid of a severe attack, but here you can have this with you without any medical help when you need the treatment. Also, there's no need for water. So you can be outdoors, you can be in outdoors activities, you can be on vacation anywhere, and you can take this anywhere at any time because you don't need water to take the medication. Another significant, important advantage is that you cannot spit it out. And this is, of course, of great value for children, that you can make sure the child gets the medication without too much fuss. And the last point here is for safety and dosing, because here you have one oral film equals exactly one dose, because we have different films for different dosages. And this is great safety concern. You don't have to mess with multiple tablets. This replaces multiple tablets, actually. And speaking more then of the allergic reactions application, we call this a rescue product, because in this indication, it's about helping when you have a quick onset, an allergic attack. And the focus area we have is called moderate to severe allergy and acute allergic reactions. And the areas are allergic rhinitis, and this is by far the biggest one. This is what is also called hay fever, hösnuva in Swedish. Next you have allergic asthma. We can also treat acute urticaria and angioedema. And in some cases we can also go even for anaphylaxis, but then it needs to be combined with adrenaline. And then speaking of market and money, of course, we have made really careful predictions, because I'm sure you know allergy is very prevalent in the world, and the numbers are increasing. For some reason, modern humanity are getting more and more allergies all the time. So we have made our calculations on rhinitis allergy, so just this large dominant indication, and it's a base of patients who have light to moderate allergy. And that's in gray. We are not targeting this one. That's an enormous market size there already, of 20 billion euros. Our target is instead the moderate to severe allergy. When it's more difficult to treat, you cannot just take your regular OTC drug to deal with it. And it may also be an acute onset, and that's when you need this film you can always bring with you. And looking just at the rhinitis indication, there are 25 to 30 million new diagnoses every year, just looking at Europe and US. And that sums up to a market size of 3.1 billion euro. And then on top, you have the most acute and severe allergy, when you get anaphylactic shock. And that's also not our focus area. And we have the treatments here, for simple pollen allergies, so to say, like I have myself. The base treatment is antihistamines, which you get without prescriptions even. And for the top segment, you have adrenaline shots developed these days. And you also have some companies working on a film delivery also for the top segment here. And you will see that later today. There's another company presenting that this afternoon. But for AcuCort, the focus area is the middle segment, where we have a very precise prescription already happening all over the globe, basically. Doctors are prescribing glucocorticoids for this treatment. And we want them to go to AcuCort, of course, and prescribe our product for these patients. And here we make a very careful estimate that we could take 10% of this market. We could calculate on having 6 prescriptions per patient over the lifetime of a patient, so that's also a very moderate estimate. And that would make 15 million films for US and Europe. And depending on the price on the film, we end up with 100 to 300 million euro market potential. And that's only for Europe and US. But we already have planning for more geographical areas. So we can certainly grow that to more. And we also are very actively working on more indications, like I already described. So we are easily doubling that in our predictions. An important topic is that this is a prescription drug, because it is for more severe cases. So you need to see a doctor to get it. And then, of course, the doctors need to be interested, right? So we have started working on this, and we have made one survey in Europe, discussing with doctors, and we had very good response there. The physicians are very positive to the treatment, to the convenience, and 90% said they would prescribe our product if they could. We made a similar study in the US, slightly different questions, and here we also had excellent response rates. We had 80% of the doctors saying that they would give AcuCort to up to half of their allergy patients to set their need. And right now we are planning for a survey more specifically in the Nordic countries for allergy and for acute onset. So that will take place now in spring this year. And then I would say this is the most important slide. This is our milestones, because this is our registration path forward. For Europe, we applied in September last year to Swedish Läkemedelsverket. And this is a very major milestone, of course, to be that close to market. Our application was based on bioequivalence studies. We don't have to do full clinical trials, since the active substance is so well known. So we just need to prove that it's equivalent to an existing drug. And here we compared it with the reference product Fortecotin, because that has a very good indicational scope. And the reply period from the authority is after 110 days, and that's in February to April this year. And we actually had the reply Monday this week. We got an enormous report back from Läkemedelsverket. And we are very happy to say that there are no major objections in it. And we don't see any major hurdles to replying, providing a lot of data, basically, to describe the process and the product. So that's ongoing now, and we published this morning, actually, in the release, because this is very important news for us. As soon as we have a Swedish approval, which is the next milestone, we will then go for an MRP, that means mutual recognition process. Then we can use the Swedish approval to very quickly gain approval in the rest of Europe. So that's our plan, as soon as we have the Swedish approval, which should be ready this year, 2020. And the other main market we are targeting is the US. It's just like many countries, it's a huge market for allergy, and it's also a very good market for convenience products. We had a response from the authority, the FDA, in September 2018, and they confirmed both the regulatory and the clinical strategy for us. We have then conducted 2 bioequivalence studies, and we are planning the application now according to a special guideline called 505(b)(2). And this is because it's not a new active substance, but it's also not just a generic. It's somewhere in between, and the FDA knows which pathway then to assess the product in. And right now, I'm commissioning a study, a computerized study, with a simulation of the response to the drug, because I want to build the knowledge in the company how the product is performing. And since we are very close to market now, of course, the next thing is to start selling, right? So we are working on establishing a network of partners. The partners could be both licensed partners taking over the product marketing and sales, or it could be distribution partners for key markets. We have evaluations and negotiations ongoing, and this is a very intense activity for myself during this year. And practically, we are, of course, preparing for commercialization. We are working on branding. We have a project for pricing. That's very important, of course, right? Pricing and reimbursement for each market. We are building a communications platform. And then we work with all the paperwork, permits, quality system, pharmacovigilance system, and so on. And I also want to mention that we really have excellent partners with us on this journey. We have very high caliber partners that have worked with us performing the clinical studies, helping understand all the regulatory requirements. Since we come into this, the application process, we have very good regulatory support for that. We have a partner for the manufacturing. The film manufacturing is done by Klaria Pharma, as you can see on the top left. And they are true experts in film manufacturing, which is very, very important here, of course. Then we have quality management and, like I mentioned, pricing and reimbursement, which has to be top class as well. So to summarize, really, the AcuCort project and the AcuCort product candidate, we are preparing it now to treat allergic reactions, to treat croup, and to treat the illness in chemotherapy called CINV. One oral film is one dose, and this can be taken anywhere and anytime, really. And that's the content. Thank you so much for listening.
Thank you very much, Ann. So, an exciting point in AcuCort's development.
It is, yeah, indeed.
Yeah.
Mm.
And like you said, you released, you had a press release out this morning, explaining a bit about the process with the regulatory authorities here in Sweden.
Yeah.
And no major objections.
Exactly. That's a formal statement, right? Because in each chapter you can have major objections and minor objections. And we have 0 major objections all the way, all the chapters. And that's very good news.
How about minor?
Yeah, minor is, we have minor, but it's not, the authority is really not objecting to anything, but they are asking for more data. And that's quite natural because oral film is a new delivery form, right? Because if it would be regular tablets, they wouldn't have to ask anything on manufacturing, for example. But here they want to know a bit more detail. I think that's perfectly normal. Yeah.
Mm. Mm. And saying that it's a new way of distributing. What are the major challenges then into going into the market with such a new product, so to speak? How much have you sort of gathered intelligence around from the other side? How?
Yeah. And I think a major challenge is, of course, the awareness among physicians. And that's why we are planning to work with licensed partners, because there we can out-license each geographical market to an expert in that area who would have a lot of inroads already to the healthcare side. Because there's nothing AcuCort can do on them. We cannot do it on our own, because that would take 100 years to gain that visibility.
And you mentioned then that the application to the Swedish authorities will then hopefully result in an approval, and that's what do you call it, a mutual MRP? What's that?
Yeah, if and when we have the approval, which should happen middle of autumn this year, we will go into an MRP, mutual recognition process. because then the European Union has already a very nice and efficient procedure to get approvals in more countries if you are approved in one. And that's why we took the strategy to go for Sweden only first to make that really work and happen without too many years passing. And then we can quite quickly copy this, so to say, for other markets in Europe.
Going then to the respective markets authorities with an
Yes,
application?
exactly. But it's one application for Europe, just pointing at Sweden as the reference.
Okay.
Yeah.
So which European markets would be next?
Yeah, we are trying to look at both big population but also good pricing. So there will be a number of both Western Europe and Eastern European countries, really. Yeah.
Do we have any questions in the audience? Yes, we do.
I wonder how about the durability? How long can I keep it in my wallet before I have to change it to a new one?
Exactly. And that's a very important question. And that's something we looked into a lot. Because for the allergy, this is not a chronic treatment, right? This is something you should have in your wallet and then use when it's time, when it's needed. And we just had stability data back, actually. We have films manufactured and being tested every 6 months for stability. And right now we had the 18-month stability, and that's perfect. That's also very good news for us. And we are aiming at having several years of so-called shelf life. We would say wallet life here. Yeah. Mm. Yeah.
Anyone else? You mentioned that, as you are in this phase of sort of beginning commercialization, which is a really hard word.
Yeah, it's a difficult word.
And you mentioned that you have ongoing discussions with possible partners and so on.
Yeah.
Can you say anything about? I realize you can't say who they are. Can you say anything about on how many different markets you're sort of spreading your activities in that factor?
Yeah, it's very difficult to tell anything, but I guess I could at least say that we are at least working on Europe, US, and then some niche markets in addition to that right now. I mean, dialogues right now. Yeah.
And another aspect of commercially that's it costs quite a lot. It can cost quite a lot. What's your financing situation like?
Yeah, like I mentioned, we just did a share issue. But like you say, you can easily spend the money, right, in this stage. That's very true. And we have... that's also why we have to balance, I would say, a successful launch right now and further work with even more applications to make sure we have financing enough.
Can't spread too thin.
We cannot spread it too thin, exactly. So we have to, be very aggressive and go forward quickly, but also with smartness, so to say. Yeah.
So going back to the application process, where now you had some response from the authorities here in Sweden. What will be the next steps going forward for you?
Regarding Sweden?
Regarding Sweden in the first place.
Yeah, now we have a 90-day window to reply to Läkemedelsverket. So I had a really heavy session yesterday with our regulatory experts to go through the first review of the Läkemedelsverket report. It's called the Primary Round Assessment Report. And that's where we concluded that we don't see any major concerns. That's why we made the communication this morning. And now it will just be hard work, so to say. We collect a lot of data, process information, even process descriptions from the manufacturing, and a number of checking on specification limits and so on. And collecting all this, and then on May 9 is the deadline for our reply. And after that, the standard process, you can have 3 review periods. This is just the first one. And if we would have perfect replies now, we could be done after the first round. But I think realistically, most companies have 2 rounds. And that's why we think we could have the approval then, something like October time. Yeah.
Very exciting.
Yes. Yes, it
To
is.
see where that goes. I would like to say thank you very much for coming here today, presenting.
Yeah.
You will also be here for a little more while. So
Yes.
Yeah.
I will stay here and both listen and be available for questions.
great.
Yeah.
Thank you very much, Ann.
Thank you. Thank you.
So I think I said earlier that we have a few adjustments to the schedule, and what we're going to do now is to actually push forward. We are 5 minutes then ahead of schedule, which is very unusual. However, for online viewers, that won't be a problem. If you happen to join us after the fact and think that you missed something, it's very easy to simply click on the little player there and go back 5 minutes in time, which is really hard to do in real life, but with online magic you can. So I'm just going to prepare the next presentation, and our next speaker is from a Lund-based company called Idogen. Anders Karlsson, welcome. The stage is yours.
Thank you. My name is Anders Karlsson, and I am the CEO of Idogen since August last year. I have a background from pharma, medtech, and biotech. I'm very excited about this new opportunity, and I'm happy to share with you what we are doing in Idogen. We are developing tolerogenic cell therapy with the aim to take care of the unwanted immune response. That is a condition you can have when you develop anti-drug antibodies. After transplantation, when you get the transplant rejection, and also in autoimmune diseases when the body attacks its own tissues. And we seek to turn off this unwanted immune response and to get a tolerance for these reactions. We have a tolerogenic cell therapy platform. It is filed for patent. We expect to get 20 new years of market exclusivity. We sent that in December this year. We are focusing on 3 major projects: IDOT, covering hemophilia, blödarsjuka, IDOT, kidney transplantation, and IDO-AID, treatment of autoimmune diseases. And that's a quite big group of different orphan indications. To better understand our technology, we need to look more into details about how the immune system works and how one of the key players, the dendritic cells, act. The monocytes are the starting points, and they tell the dendritic cells either to be tolerant or intolerant. And dependent on the subject, it can activate immune response and then start to attack the subject. It can also downregulate the immune system and also then tell the body that immune system that this is okay. There is a lot of things for the immune system to sort out. It can be virus, bacteria, cancer cells. It can also be pharmaceuticals. It can be food. It can be transplants, and it can be the patient's own tissue. And as long as the system works as it should, there is no problem. But in the conditions I explained before, there are situations where the immune system doesn't act as it should. Our tolerogenic cell therapy takes care of that and reprogram the immune system to tolerate the pharmaceuticals, the transplants, and also the body's own tissues. Our tolerogenic cell therapy is tailor-made for the patient. We use the patient's own cells. We collect the monocytes from the patient at the local hospital. We send them to a manufacturing site where we introduce the monocytes to the disease-specific antigens. We add a tolerance inducer, and other critical substances, and we develop a dendritic tolerogenic cell. And that is specific for this disease. After that, we send back to the local hospital, and we treat the patients over 3 to 4 times bi-weekly with the aim to create a tolerance for this specific disease or condition. Over this time, the body then induce, or the tolerogenic cells, dendritic cells induce regulatory T cells, which is very important to keep the long-term tolerance for the pharmaceutical and for the transplant and so on. The first project is IDO8, and that targets hemophilia and patients with antibodies being treated for the disease. Standard treatment is factor VIII, which is a clotting factor, and that is actually what you miss when you have the bleeding hemophilia A. And you see the different stages here. This is a bleeding, normal function, but when you develop these antibodies, the system doesn't work as it should. The normal treatment is factor VIII, replacement of factor VIII. But in 25 to 30% of the cases, the patient develop inhibitors or antibodies. That you can treat. In some of the cases, you usually give higher doses of factor VIII, but that is quite a difficult treatment. You do it over 6 months, up to 18 months, and it's quite costly. The cost for these patients is around $140,000 a year, and if you need to treat for the antibodies, it can be up to $1 million. IDO8 aims to reprogram the immune system to tolerize factor VIII and regaining its effect, so you get a normal clotting system after that. It is a huge market. 150,000 globally has hemophilia type A. We have in our target market only focused on market where you have a healthcare system that is able to finance this kind of treatments. So we are aiming at North America, Europe, and Japan. This market has around 56,000 patients identified with hemophilia. Around 1/3, 18,000 patients, develop antibodies and get then treatment for that. Our first target is around 1/3 of that market, where you not really have a treatment available today. But for sure, if you can create a tolerance for factor VIII, this could be expanded to also include the group above. The market is growing relatively fast. From now and 10 years ahead, there will be an annual growth of around 6% in the most likely scenario, and has a value at that time around $20 billion. We have a clear development strategy for IDO8. 2019 has been a lot of preclinical preparations. 2020 is to start to prepare for clinical trials. 2021, the first patient will be treated with our tolerogenic cell therapy. So it's quite soon. A major step forward was performed during this year. In October, we announced that we have a collaboration with Radboud University Medical Center in the Netherlands. They are specialists and have done that for over 20 years to manufacture cell therapy based on dendritic cells. So we have now moved, as we speak, our process developed in Lund to the Netherlands, and they are now preparing to scale up for GMP product that can be used in the clinical trials later on. Next project is IDO-T, and IDO8 paves the way for that. what we learn with IDO8 will for sure be used in the clinical development of IDO-T. Kidney transplantation is also a huge market. 80,000 patients perform kidney transplantation every year. Patients on performing transplantation has lifelong treatment with immunosuppressant drugs. And there is a need actually to lower the doses and be able to get rid of the unwanted effects of the immunosuppressant drugs, which is high risk for infections, high risk for cancer, but also a direct negative effect on the kidney as such. Our treatment aims to educate the immune system to tolerate the new kidney in the body and tolerate that, and then potentially lower the dose of immunosuppressant drugs or potentially do not need. And if we look into what happens is that we pre-treat the patient with cells before transplantation. There we create the T cells that supports the long-term tolerogenicity of the new kidney. This is a group with very high treatment costs. Before transplantation, they are on dialysis with high cost, but also a low quality of life. They need to visit hospital, most of them 3 times a week. Also, the cost for transplantation, but also after, is relatively high. But when you have them on transplantation and you have a functioning organ, that is a good investment for the patient for sure, but also for the healthcare system as such. If you once again look at the market that I've indicated before, North America, Europe, and Japan, we have there a target market of around 45,000 patients. 26% of them on these markets perform transplantation with a living donor. And that is the case when our system can work. You can plan in advance for the transplantation. You schedule the transplantation at the time when the patient is prepared for it, and you get a better outcome. And you can then also have everything ready when you have the cell therapy ready. The development program looks very much like IDO8, but it's sequenced slightly later. We will start do things this year. We will continue in 2021 and aim to start clinical trial, if everything goes as it should, in 2022. So it's a delay between the different indications, but it also, resource-wise, we need to use what we learn for the first indication to be able to move forward. The next indication, autoimmune diseases, that's a broad group of patients. There are identified more than 100 different diseases. Many of them are orphan with high unmet medical needs. Some of them has a strong fit with our platform. That means we have a known antigen. It's T-cell driven. And then we need to find the ones that fits best to treat with our technology. This is the next step after the 2 initial projects that we are planning more in detail for the time being. And then the question is the market ready for cell therapy? Are we willing to invest such in the healthcare system today? If we look into different stakeholders, MIT in Boston estimates that 50 to 30 cell and gene therapy products will be launched the next 5 years. FDA estimates approval of 10 to 20 cell and gene therapy products within the next 5 years. And the market value is estimated in the most likely scenario to be around $40 billion 2024. On this graph, you see the accumulated launched cell and gene therapy product estimated for the years up to 2030. Another way to see it is cell therapy is not really new things. I mean, it has been performed since maybe 10 years back, maybe also earlier, but at a very early stage. It was more research. Today we have the first product launched a year ago, CAR-T product from Novartis, and everyone expects that this market will grow for a few patients and severe patients, but it will help a lot of patients that we now have the ability to treat in the future. Another angle on this is that this is quite expensive development, and a lot of big pharma company is now entering the market, screening, looking in, finding partners, acquiring small company with innovations. But there is a fit that the small company needs the structure from the big pharma companies to build the competencies to launch the pure product for the future. Our idea is to bring our project so far so that we see the first efficacy in patients. Then we need support to do the next step and further develop the products. As I said before, we have a platform in for patent filing. We will have 20 years of market exclusivity. We are aiming at IDO 8, which gives us around 6,700 patients in the target market. Kidney transplantation gives us more than 11,000 patients every year. And the autoimmune diseases is quite big, but we have not really made the estimations yet. The future is bright, and the future belongs to cell therapy, I would say. Not to all patients, but those who really needs it. We have established a novel and innovative cell therapy technology. We have an efficient and patentable tolerance inducer that has been identified, and we are addressing unmet medical needs that opens up for attractive reimbursement possibilities. We have clinical readiness next year, so 1 year until we have the first patients in phase 1, phase 2 clinical trials in hemophilia. In order to continue to develop in a fast pace, we have also communicated a new issue. It will be performed during March. One current Idogen shares gives right to buy 1 new share. It's a 1-to-1 price, 0.6 Swedish kronas. Subscription period, March 2 to 16th. And it's guaranteed to 88% from a consortium, but also from management in the company, as well as the board of directors. We have the financial advisor from Erik Penser Bank. And the proceeds will be used to upscaling the production. That costs around 9 million, is estimated to that. Preclinical evaluations, mainly on transplantation. And we also want to strengthen the working capital in order to finance the process until we have the clinical trial up and running. And that's around 9 million. So we have cash after that, also including a horizon funding until Q3 2001, assuming that we make success in this new issue. Thank you.
Thank you very much, Anders. Yeah, you said cell therapy is the future. It's a very exciting future, I would say. So where Idogen is standing right now, you had some trouble last year. You had to sort of start over, you can say, in a sense. And now you're aiming for going for the clinic. And what will be the biggest challenge for you? I mean, I know you mentioned production, but what is the biggest challenge for you going forward, you would say?
I would say that, I mean, there is always challenging when you're doing things you've not done before. You're saying that we had problems early last year, and that's correct. We developed a new tolerance inducer in less than half a year. And you cannot do that without having the experience as we do have in the company. The company was started in 2007 out from the university academia in Lund. So there is a lot of knowledge within the organization. We have also scaled up the organization quite a lot the last 2 to 3 years. And we have a strong organization in place now. So yes, there is a lot of things to take care of. And you're doing things which is on now. However, we are very happy to be where we are for the time being.
Confident. I should turn to the audience. Any questions in the audience? As of now? No. You mentioned the ongoing, or so soon to start financing round, about 29 million after costs, and
Before cost.
Before cost, sorry, and about a third of the capital will be used, or even actually a little bit more than a third will be used for the production process. Correct. And then you also mentioned then the ongoing operations. Can you specify a little bit about that?
Yeah, I mean, we have this autumn accelerated quite a lot in the organization. A lot of new things has happened, and this also indicates a higher speed to work towards the objectives that we have set up. I mean, it's important to deliver on time and be able to start the clinical trial as soon as possible.
You also entered an agreement with a partner, production partner.
That's correct, in the Netherlands, and for sure we assess them very well before we enter into this. And they have a great experience been doing this, and they are global leaders in manufacturing cell therapy. However, I mean, we have been the one to develop the technique, but they have the ability to scale up in a GMP facility, which is important then when you start to perform the clinical trials.
And about the upcoming clinical trials then, how big do they have to be? If you give an estimate about how many patients would you have to include?
First, we start to do the clinical study on patients. We can do a phase 1 safety study on patients. We are using the patient's own cells. So that's the first step. And then we want also to include efficacy measurement. We aim at 9 to 12 patients preliminary. So this is the first test that we have a clinical success and that we see efficacy. To take it a step further, then you for sure need more patients. But we say that having this data tells us that we have a lot of interest within this kind of technology. So our objective is to bring it there, look into what kind of options are out there, and see how we in best case can go forward from that. And that is also the same for transplantation. Same steps into patients and then assess.
And given that cell therapy in general is a very sort of hot area, have Idogen experienced any certain interest from external players, or is it too soon, considered too soon?
There is an interest. Then, as always, people are saying that we want to see more. But even if you want to see more, you want to build up relations. And yes, I mean, there is an interest.
Any questions already? Yes.
In this hemophilia, there is one protein that you need to induce tolerance against. In the transplantations, will you need to induce tolerance towards several proteins for it to work?
Yes, and I mean, that is what we need to assess also, how one actually do that. And we are looking into if we can prove that in animal models before starting up, yes.
Questions? If not, then we'll say thank you, and it's time for a little bit of a lunch break. So thank you very much,
Jonas.
Anders.
Thank
You will be here during the lunch as well?
Yes, I will be here.
Okay, great.
Thank you.
Thank you very much. We will take a short So, welcome back everyone for our third and final session for today. We have 4 more companies to go, and I'm pleased to say welcome to the next speaker, Jesper Wiklund of Klaria Pharma. The stage is yours.
Thank you. Yeah, okay, so as mentioned, my name is Jesper Wiklund and I'm CEO of Klaria. So Klaria was founded in 2012 and the company has been listed on the stock market since 2016 here in Stockholm. We have a patented and proprietary technology platform which forms the basis of all of our drug development, and I will be telling you about that platform today. We have 2 late-stage assets in the company that are the key drivers of value in our company right now, simply due to the fact that they are the furthest advanced and the closest to the market. We have a number of pipeline assets that are earlier, that are also very exciting from a commercial point of view and from a medical point of view, but they're earlier, so I will focus today on the 2 late-stage assets, which are sumatriptan, which is a treatment for migraine, and cannabis, which is exactly what it sounds like. It's marijuana, cannabis, and in these 2, we are formulating both of these products into the film. With sumatriptan as a treatment for migraine, our projections, and we'll go more into detail about those projections, but our market projections for the sales potential of our compound, of our product in the United States and in the sort of 7 or 8 countries in Europe that pay premiums for medicines, reaches $250 million in peak sales potential for our molecule. So that's not the market, but that's our molecule. For cannabis, we are launching into $1 billion. Well, there are multiple markets in cannabis, but each one of them is worth billions of dollars. So both of these assets are addressing very significant markets and have very significant sales potential. Very importantly, I recently joined the company. I joined the company in November of last year, and as I was getting to know this company and as I myself was trying to understand the technology and evaluate the technology, I took a lot of comfort into the fact that Klaria has already a partnership with Mundi Pharma, which is a global big pharma player that has thousands of scientists, people who are working for them, who are obviously, highly educated professional people with close to unlimited resources in terms of what they have at their disposal to evaluate possible collaboration partners. And they, after having surveyed the entire world really for the best drug delivery technology for their products that they had an intention of developing, they chose Klaria. And they chose Klaria after having done a very thorough due diligence where they had a team of 15 people come over here to Sweden to go through everything, to look at everything, to look at the patents, look at the technology, the manufacturing, all the clinical and preclinical data that has been gathered to date. So what I'm getting at is this is a very strong validation. And for myself, and I imagine all of you, as we try to understand a company, you can look at the homepage, you can maybe look at some analyst reports, you can look at a few different things, but really it's very hard to really understand, does this company do what they say they do? Can they actually deliver on the technology that they have? And the fact that an international pharmaceutical company has actually come to that conclusion after having had their team of experts looking at this company for months and months, gave me, and I believe should give all of you, great comfort that this is in fact a technology that really has very strong legs, and it's a technology that actually does what it says it does. We also have commercial validation through these films, which I handed out to you, or to many of you previously. We'll get to them in a second. These films are for sale in Swedish pharmacies, and they're formulated with vitamins. So this company, Nutrinovate, is a licensee of our technology. So it's our exact same technology, but rather than, as we do, formulating pharmaceuticals into these films, our licensee, Nutrinovate, has formulated vitamins into these films. And we consider this commercial validation in the sense that this actually shows that you can manufacture these things, you can package them, ship them, distribute them, and sell them and make a good business out of it. And that's something that very few companies that don't actually have a product on the market, as we do not, but very few companies can actually say that, we know that this works as a business. But through our relationship with this company, we can actually make that claim. Okay. Just one more word about background, and then we'll get to the heart of the presentation. At the beginning of the year, last year, the company was led by Scott Boyer, who was the CEO. He's also a founder. He's the sort of the scientific heart and mind of this company. Over the course of last year, we expanded the management team to really help transition the company from being a science-driven, research-driven, development-driven company, which we still are, but actually helping us transition now to being a much more of a commercial company, which is the next stage that we're going into, and it's a stage that we also look forward to with great enthusiasm. And so in addition to myself as the CEO, we also have added Scott DeMarcado, who is as a business development, and he has focus on the cannabis business development primarily. And we also added Mark, and Mark is a professional in manufacturing, and he has over 20 years of manufacturing expertise in some of the leading companies in the world. And this manufacturing expertise becomes incredibly important the closer we get to the market, because if you survey the landscape of disasters in our industry, a very significant portion of them actually happened because there's something that went wrong with manufacturing. So we're taking manufacturing very, very seriously. We're getting the best possible people that we can. We're working with the best possible partners that we can to really try to make sure that we do everything right in manufacturing. And I myself I've been a CEO of a biotech company previously. I've also worked for an American private equity fund for the last 5 years. Scott DeMarcado also has very significant experience in sales and marketing. So overall now we believe that we have a team together with Scott that is highly qualified and that can really help execute all of these plans that we have and for the future that I'm also going to tell you about. So let's get to how it works. So if you now, everybody take out this vitamin film that I handed out earlier, if you like. This is, again, it's nothing dangerous. It's vitamins that are being sold. So if you want to, you can go ahead and open it, and you can take out the film, and you see the film here, and you just put that film on the inside of your cheek like this. And I see that nobody's doing that, but if you want to do that, go ahead and do it. I think it's if you don't have one of your neighbors should, because I try to hand out as many as possible. Anyway, here's one. Do you guys want to try it? He wants to do it later. All right. Anyway, here, you can try these if you'd like. Let's hand those around. And the reason I'm handing these out is that it actually becomes very obvious as soon as you put this film into your mouth what the key benefit of this film is, and that is that it sticks to the mucosa, to the inside of your cheek. And this is pretty much the only substance in nature that actually sticks to the mucosa, which makes a lot of sense if you think about it, because one of the key purposes of the mouth is really to make sure that stuff doesn't stick to it, and then it actually goes down into the stomach, where whatever you put into your mouth, the purpose is to get it into your stomach. So things don't stick to the mouth or to the inside of the mouth. And that is the challenge when you're trying to achieve transmucosal delivery, because what you need is you need to have the film stick to the inside of the cheek and then stay there. And during this time that it stays there, there's a high concentration of drug in the film, and that concentration, that drug, transfers across the mucosa into the blood. And that is indeed what happens with our film. Now, what separates us from pretty much all other technologies that claim, because the idea of transmucosal delivery is not a new one. It's a very old idea, and it's an idea that has a lot of appeal because, for obvious reasons, if you can avoid the stomach, there are many, many benefits to that. But there are many other delivery technologies that have tried to do this, but they don't work. There are sprays, there are tablets that you're supposed to put on your tongue and leave them there, etc. And what happens with all of these other technologies is that you put them in your mouth and they mix with the saliva and they melt. And then so you have this little pool of melted substance in your mouth, and you might have it under your tongue for 5 or 10 seconds, but then you're going to swallow it. And that's just human nature. If something's in your mouth, you swallow it or you spit it out. I mean, those are the two kind of options you have. And so while you might get a very small proportion of the drug being transferred across the mucosa, the vast majority of all of these other technologies, the vast majority of that drug gets swallowed, and it actually becomes oral delivery. And that does not happen with our film. Those of you that took it, you can feel that it's still sitting here. And so what this allows us to do, and sort of the proof, in addition to the fact that we've convinced a big pharmaceutical company that we're actually able to do this, kind of the other proof that we're really able to do this is that we are actually focusing on putting molecules into our film that are not orally available, meaning if you swallow them, the body does not take them up, right? So if what I'm now telling you was not true, our entire strategy wouldn't work, right? So this is sort of the key thing to understand about our technology, and it's a pretty big claim, especially for a small company based in Sweden that today is not tremendously valuable, et cetera, but it really is, this is the key thing that you need to understand. And if you understand this, then our entire pipeline and the products that we have start to make sense. So let's now talk about the two products. Well, first, maybe I'll just a moment about our strategy, and then we'll talk about our two lead products. So given this feature that we have, that we actually achieve transmucosal delivery, we've then decided to focus on developing drugs where this feature results in the greatest benefit. Because if we create a lot of value, if we create a lot of benefit, our feeling is that patients will benefit from that, and we and our shareholders will also benefit maximally from that. So we look at the sort of addressable universe of compounds that we could put into this film, and we ask ourselves, first of all, are these today primarily or only used as an injection or a nasal spray? And if they are, you're sort of in this green zone here. And then you ask yourself, so we ask ourselves, is that a problem? Because sometimes it's not an issue. But where that is an issue, that's where we end up in our sweet spot. And this is where we want to focus our time, effort, and money and talent. And here we have the products that we have in our pipeline. We have adrenaline, which is for anaphylactic shock. We have midazolam for acute epilepsy. We have naloxone for opioid overdose. We have sumatriptan, which is a triptan. We have other triptans as well for migraine. And we have ketamine for acute pain and depression. Now, all of these are tremendously exciting. We only have 15 minutes here, so I've decided to focus on the two most advanced ones, which are the ketamine and also the cannabis, which is actually not on this slide because we've created a daughter company that addresses the cannabis opportunity. I'll tell you more about that in a second. But any of you want to learn more about this, you can either come talk to me afterwards, or you can also look at our homepage, and we have more information about all of these programs. we can skip that. Okay, so let's talk about sumatriptan for migraine. So as you may or may not know, but sumatriptan is an old molecule. It's been around for a long time. And there are a number of, well, there's a tablet, there's a nasal spray, and there's an self-injection pen. Those are the three ways that you can today use sumatriptan. And sumatriptan is the sort of first-line treatment for serious migraine. So why do we think that our film makes sense to develop in this context? Well, 80% of all of the patients that suffer from migraine also suffer from nausea. That means that you wake up at 3 o'clock in the morning, you've got a migraine, you need to take your medicine. If you take a tablet with a glass of water, in 80% of the patients, it's quite likely that 20 minutes from now, you're going to throw that up. Or 10 minutes from now, you're going to throw that up. The throwing up part is not so bad. The bad part is you actually throw up all the medicine you just took a few minutes earlier. So, you're not getting any drug into your system and you're not getting any pain relief. So because of that, people then use nasal spray or the injection. The problem with the nasal spray is, and everybody who's ever taken a nasal drop, if you go on an airplane or something, you know this, you take a nasal drop, and actually what happens is a little bit of it it's supposed to be mucosal delivery because you have mucosa here in the nose, right? But what actually happens is some of it sort of passes by the mucosa, but most of it ends up falling back the back of your throat, and you swallow it, and you're back in the stomach again, right? So it's oral. And if you throw up, you've got the same problem. You're not getting drug on board. So then people take injections, but the problems with injections are many. I mean, it's nobody likes to take an injection. You have to shoot yourself in the thigh. It's never pleasant. It's not pleasant at 3 a.m. when you have a brutal headache. It's painful. You can get reactions at the injection site. You can get infections, etc., etc. It's in solution because it's a pre-injection pen. So the stability is not good because anything that's in solution has a shorter stability. So it becomes expensive. You got to have it in your fridge. You got to keep buying new ones. A bunch of problems. So this leads to this explains this statistic. 79% of all migraine sufferers have said that they're willing to try a new medication if there was one. I have 3 minutes. Okay, I got to go a little quicker here. So this is basically the situation that I'm describing. People bounce around. They can't find a drug that works for them. So this is what we call an unmet medical need, right? And we believe that our drug can really fill this need because it has basically all the advantages of an injection, meaning even if you throw up, it's not a problem, but it's not an injection, right? So if I move on, you can also please talk to me about this afterwards. We believe that this is going to be approximately a $250 million per year sales for our compound. And we believe that based on an analysis that we've done of the key territories in Europe and in the United States and considering the market dynamics. If I move on quickly to cannabis, as I said, we've created a separate daughter company that does this. To go through this, basically there are two ways to take cannabis today. You can smoke it or you can eat it. Smoking is the best in terms of having an effect that is fast, and also in terms of actually having a proportion of the drug that you take into your body sort of get out into your blood so you can have a medical effect. The problem with smoking is that it's very bad for you from a health point of view. It's illegal almost everywhere, and most people consider smoking no longer acceptable. It's socially stigmatized. So therefore, the entire cannabis industry, which is a $1 billion industry in the United States and in Canada, think that edibles are the future. The problem with edibles is that it takes almost an hour before you get an effect. And the uptake is very different. So if you take, for example, a gummy bear, cannabis very often comes in gummy bears. You take a gummy bear 1 week, you get a certain effect. The next week, you might get a dramatically different effect, depending on what you've eaten, if you've eaten, if you've slept, if you've not slept, because the stomach is just an incredibly sensitive organ. So again, here we have basically the best of both worlds. We have, and we've shown this in preclinical trials, we're now about to start clinical trials with this, but with our film, because we actually transfer across the mucosa and we don't go into the stomach, you get an effect in sort of 15 to 20 minutes. It's not as good as smoking, which is 10 to 15 minutes, but it's pretty darn close. And it's way, way better than eating, which takes up to an hour. And so we are now in advanced, or we are in discussions with a number of companies in the United States, in Canada, in Jamaica, in Japan, that are manufacturing and selling various marijuana formulations in the territories where this is legal and commonly accepted. And everybody that we speak to, they very quickly understand what we're saying, and we're meeting with a lot of enthusiasm. So we're very hopeful that we're going to be able to conclude some sort of agreement with one of these discussion partners and actually start manufacturing. And the beautiful thing from our point of view about this is, if you get into what's called the recreational segment, there's no regulatory hurdle, so you can just start making it and selling it. It's like making a vitamin. It's even less than making a vitamin. And so here we could be on the market in 2021. Very, very quickly. We did a lot of very important things, both from a corporate development point of view and from a pipeline point of view, in 2019. So we did a lot of things. However, we didn't really tell anybody about that, and we're aware of that. So the stock market doesn't know about that. You don't know about that. And so therefore, our share price doesn't reflect it. But we did a lot, and we have a lot of plans for 2020. One of the key ones is the last one here, that we're going to get better about speaking to the stock market. This is the first presentation like this that I do. I've now joined, and I'm putting a great focus on telling our story to investors this year. We last week launched a new website that we hope will be much more illustrative and explanatory about what we're trying to do. We're also speaking much more with potential partners. So we think and hope that 2020 will be a year when we achieve a lot in terms of the operational side, and also hopefully that we achieve a lot in terms of our communication with shareholders in the stock market. So am I completely out of time? Yes. Okay. So we have two late-stage assets. One, if things go well, and we hope they will be ready for launch in 2022. It has a $250 million sales potential. That's an asset that should be worth the same amount when it's approved, 10 times what we're worth today. Similarly, with cannabis, we have the billion-dollar markets. We have a differentiated product, which is patented and which is completely new, that we have great hopes for. So we hope between the two of these, there's going to be a very interesting journey for ourselves and our shareholders in the next couple of years. Thank you.
Thank you very much, Jesper. You actually did very well in using up all your minutes.
Thank you.
So we don't really have time for Q&A here. I'm sure that there are lots of questions, though. But I'd like to congratulate you to be the first, I think, company that's ever been to BioStock Live that actually brought samples of your product.
Yeah. And
I know that Stefan from Endovab had a device with him, but that's not as easy to try out.
No.
So maybe in a year or so we can try it with some other compound inside it.
Yeah, if we need a doctor then to diagnose people and give prescriptions, but or if we do it in California, we could all try cannabis.
I'm all for going to California, right, so let's do that.
Okay.
A warm hand for Jesper. Yeah. Thank you very much.
Thank you. Thanks.
And then that means that we're going to have to rush forward in our program. And let's see if I can be of assistance here and do the technical stuff. Next speaker is Marcus Bosson, CEO of QuiaPEG. Is that
QuiaPEG, yeah.
QuiaPEG, and Marcus, the stage is yours. There you go.
There you go. Yep. Thank you very much. Well, my name is Marcus Bosson. I'm the CEO and co-founder of QuiaPEG Pharmaceutical, a biotech company based on a proprietary drug delivery platform. We are located in Uppsala, former Pharmacia area. We have our lab and offices there. So our drug delivery platform is called Unicleaver. It's chemistry combined with polyethylene glycol, and I will come back to you in a minute what exactly we do. So we are based on a scalable business model. So we usually say we are not a one-trick pony, one project, and we kind of stuck to that project. We have a quite broad platform which is scalable. So in the future, we can actually get short-term revenues in form of licensing of a drug delivery platform to pharmaceutical companies developing new pharmaceuticals where the half-life is a problem. We could extend that half-life. And in parallel, we are working on building our own pipeline of biobetters, utilizing already approved drugs and making it a longer half-life. That means instead of once daily, maybe once weekly. That's what we do. So, is there any money in this kind of business with drug delivery and half-life extension? Well, we have a benchmark company. It's called Ascendis. It's based on German invention, Danish entrepreneurship, and excellent business craftsmanship. And it's listed on Nasdaq in New York City. And actually, I checked the market cap today. It was founded 2008, and it's even $7 billion. So it's one of the top 100 companies, biggest pharma companies in the world today. And they have a similar technology, increasing the half-life with a releasable technology platform. And they have succeeded now in a phase 3 clinical trial with human growth hormone, where instead of once daily, you take it once weekly. So definitely it's interesting. As you can see, they did some years ago out-licensing to Sanofi, Genentech, and United Therapeutics, and it totals up to about 60 million euros. And that was preclinical, only for the platform, nothing regarding specific projects. So, Unicleaver, how does it work? Well, as I mentioned, it's a prodrug. And what does a prodrug mean? It means when you couple our technology to a molecule, a peptide protein or a small molecule, it kind of protects this drug. And then it's released by pH. We all have the same pH, 7.4. So depending on how you design this activated PEG, which is proprietary, you can have different cleavage time. So, for instance, 50 hours or 10 hours or 80 hours, depending on the substance. It protects, it's biologically inactive. And then with the pH, it starts cleaving. And once it has cleaved, it's fully native, the biological effect comes back. And polyethylene glycol, PEG, maybe you have heard of a golf peg, but this is chemistry. It is widely used, has been used for 30 years. You have several blockbusters utilizing this PEG, Cimzia, Neulasta, etc. Basically, literally, millions of people are using PEGylated drugs every day. You have all used PEG today, either by creams or food or something. It's widely used. So that means it doesn't have side effects, potential side effects, but it has been tested. So it's well known how it works, pros and cons. So what you then take is the drug, and then we have our proprietary technology called Unicleaver. So you connect it together. And then you can design this Unicleaver, depending on the half-life, how long it circulates in the blood, a different trigger from this library. I will come to that picture. And then when pH kicks in, you get the split, the cleavage. So you have a PEG for itself, and it's cleared through the system, and you have an active drug which can do the job. So this is a summary of our invention. So it's in the middle you have a phosphotriester with a trigger, which you select from this trigger library, where you have different cleavage time. So you can tailor-made this release speed. Then you have a polyethylene glycol and the drug. So by combining this, you can tailor-made the half-life of the drug, increase the half-life of the drug. And we usually say it's universal. It's triggered cleavage by either enzymes or pH. And enzymes could be very important in the development of antibody-drug conjugates, which is a big area in cancer oncology today. It's efficient, so you have a complete cleavage. You don't have any residue left on the pharmaceutical, which is good from a regulatory perspective. It's predictable, it's controlled, and we have a growing patent estate. And now I see that actually BioStock used my old presentation, so I have an updated version, unfortunately, but I will do my best. So, and the clinical advantages is actually fewer doses, increased patient benefits, and lower cost to society. With stable blood concentrations, you can most likely reach a better, less adverse effects. And regulatory-wise, you can use this called the pathway called Section 505, which is you can piggyback on what's already out there in the filings, documentations of an original pharmaceutical product. So we call the biobetters. Why is it biobetter? You have all heard about biosimilars. Biosimilars, that's supposed to be similar to the original drug. When it's small molecules, you call it generics. In bio, you call it biosimilars. Biobetters, that's supposed to be better than the original product and the biosimilar, which means that you add something good for the patient. That means you can maybe patent it also, protect it. You can maybe also have a flexibility of getting a premium price instead of competing with the lowest price possible. And also you have some other goodies, data exclusivity, etc. So we selected some time back how to show this, all what we're saying, how to show that this is really true. So we went through different potential pharmaceuticals, and we actually came up with this as a model project, pegylated liraglutide, and it's a peptide. And it's a blockbuster drug for Novo Nordisk. The patent is soon expiring. It's about $4 billion annually. It's administered daily by injection, and also a key thing is that 10-15% of the patients, they stop taking it because of side effects, nausea, vomiting, not feeling well, to put it simple. So what we are trying to do is actually to develop a biobetter, to be administered once weekly, and potentially with less side effects compared to the original. And we have filed a patent on this, so that expires 2038. More patents will follow regarding production, etc., so we can probably extend it. And it's also a possibility to file this under Section 505(b)(2). And what's really good about the potential of less side effects is that, as I said, we have this protection of the original drug and its controlled release. That means we don't have that spike when you inject it the first time. So you could have this more steady state of release of liraglutide or any other peptide, which means that potentially it could be less side effects, and that's very good sales argument. So, unfortunately, I had an updated version, but that doesn't seem. So I will go over to the pharmacokinetic pilot study. We actually had 2 press releases. One was last week with that we had finished the development of 2 analytical methods. It was together with a global CRO, very well renowned. It has taken some time, so we made one for liraglutide and one for the conjugate, the pegylated liraglutide. And we need that to measure the different levels moving forward also in preclinic and eventually clinical trial. And then we released a couple of days ago a press release of a pharmacokinetic pilot study. So we had a rat study. They were injected subcutaneously and intravenously. Subcutaneously means under the skin, and IV means into the blood. And the advantage with sub-Q administration, subcutaneous administration, is that you don't have to go to the doctor to administer it. So we measured the pegylated liraglutide, and then we had one control group with liraglutide. And what we see is actually the liraglutide is released as expected from the conjugate. It reaches the bloodstream when injected subcutaneously, and for you, well, it doesn't sound much, but it's actually an important milestone to tick that box that we could inject it subcutaneously and get this controlled release. It's actually a milestone. And after the administration, we can see that the plasma half-life, that the half-life of this in the blood is significantly extended compared to direct administration of liraglutide. So we're really onto something here. And this not only validates this project so far, it also validates the Unicleaver technology platform, that if it works with this peptide, maybe it also works with, supposedly, with other peptides and proteins. So it's really very good data. All right, I will quickly go through the company, the management, and I'm the co-founder, and Marek Kwiatkowski co-founder, and we have a board with a chairman, Steen Kreuer, who used to be the chairman of the CEO of AstraZeneca in Shanghai, and actually built it through 6 years and made it to what it is today. We have a growing patent estate. We have 11 approved patents. We also have a good shareholder structure. Neuenburg Investment Partners, which recently, I think, joined Episurf shareholder community. And also, you can see that the founders, we have a lot of skin in the game, and I think that's one important signal to you as potential shareholders or shareholders, that we are really dedicated financially also to this company. So the ongoing and planned activities, we have announced that we will soon have proof of concept results, and that's ongoing. And those results will be very, very important for us. It's a big study. We have pegylated liraglutide, 2 doses. We compare it, benchmark it to liraglutide 2 doses and to the semaglutide 2 doses in which is Novo Nordisk once weekly peptide, which also have the same adverse effects as liraglutide. So, and then we have a control group. So it's a food intake study. So you have the rats, they're running around, and the control group, which has not received anything, they eat and are happy they eat during the night, actually. So for a certain amount of time, you measure how much they eat, and also you weigh them once daily. And they are supposed to, with this liraglutide, pegylated liraglutide and semaglutide, to lose weight compared to the control. We'll see what the results will turn out to be, but it's really a proof of concept study for us. And that's going to be announced soon. In parallel, we have a CMC aspect ongoing, and I agree with a previous speaker that contract manufacturing or manufacturing is actually one of the key things for every biotech company. If you have any delays, if you have any cost increase, it's due to that, I can almost guarantee you. So that's ongoing. We have a very good partner in Germany. It's kind of a firm where to go to when it's pegylation. We have planned now additional preclinical studies and also the tox studies. And last but not least, we have ongoing serious licensing discussions in China with a potential partner for this pegylated liraglutide. There is a big interest in China for GLP-1, which is diabetes and oncology, and new technology platforms. We have been to several conferences. It's huge interest, and they do have money. So, but they don't have exactly that kind of technology. So now they are focused to new inventions. And also we have, as you know, some ongoing collaborations, testing of this Unicleaver platform. That's progressing, and we also have other ongoing discussions. And with these data coming out now, I'm pretty sure that we will enter a very important phase in those discussions. So that's where we are with the business development. And let me just finalize why invest in QuiaPEG. Well, Unicleaver is a versatile platform, limited competition. I usually say it's about 5 worldwide companies, and a couple of those, they don't out-license their technology. So I think we have a good chance of, if we reach that level of publications, awareness of what we do and who we are, I'm pretty sure we do have a very important position in this market. And parallel, we are value creating by building a pipeline of bio-betters. That means limited risk for the investor because it's known pharmaceutical drugs. And we have this also reduced commercial risk because it's really accepted the pegylation area, utilizing pegylation as a sort of a form of half-life extension. And last, it could be also interesting potential acquisition target for a big pharma company. As I mentioned before, we also have technology for ADCs, the linker technology, and we have this for biobetters and also for new molecules. So a big pharma with a big pipeline, they could really make use of this platform. And by that, I thank you for your kind attention.
Thank you very much, Marcus. Sorry about the mix-up with the presentation.
Okay.
I think you got a fairly interesting message through anyways.
Okay.
So something that caught my interest was this, when you work with creating a biobetter, you said that there's a low risk and it's quicker than to develop a brand new drug. And but what are the differences between to develop a biosimilar towards a biobetter? Is it not a bit longer process?
I think, yeah, it's a biobetter because you insert something which makes it better. So of course you have regulatory-wise a little bit more burden than a biosimilar, which is supposed to be about the same, no better, no worse than the original drug. So, but I mean that in that field you have plenty of big competition. Big is beautiful, and then you need to have a marketing setup and be aggressive and put a lot of time and money into marketing. Biobetter, we achieve something which is better, which means that if you have original product up here, price-wise, you have maybe, and then after it comes off patent, it will come down, but often, like Novo Nordisk, we have a semaglutide, which is still up here, price-wise, but the liraglutide might be down here. And because we will be compared to the semaglutide, which is the once weekly, we have a flexibility in between to put us price-wise. So we don't have to compete with the lowest price possible. So that, but yes, it's a little bit more work and a little bit more investment, but I think in the long run, when you don't have to compete with the price, you can make it back quickly. So.
And then looking from the other perspective, then working towards finding a biobetter for one of those, blockbusters, really. The original owners, like Novo Nordisk, for example, they should be monitoring your activities fairly closely, I would say. Are they possible takers for your project?
That's speculation, but
Of course.
yeah, of course. But maybe the big ones, they have not invented here attitude. I don't know. I don't know how familiar just specifically Novo Nordisk is, but it might be so. So, but you have several others, of course, to take us to this. So, and I should point out that maybe we will take this through not only a tox, but maybe a phase 1/2A, so we can measure the efficacy in humans, because that would be tremendous value for all other potential targets we have. So,
Yeah, and about that, it seems like this platform technology, if you will, has a broad potential. And are there any? You mentioned that a potential acquisition target, sitting on an asset like this. What would make you take a decision that you would actually let someone buy you when you have that potential?
I don't know. It's a
huge bags of money. Or
yeah. Well, you can say today. I don't know what is it. 100 million Swedish krona, give or take a few, and that's $10 million. And if you ask yourself, $10 million, how much is that for a big pharma company? Maybe not so much. But I'm not speculating about the stock price, but. I can see that in the future, this really now when we are gathering hard facts, that this could be really a good target for acquisition and a clean cut. And I expect us, a co-founder, put a lot of time and effort and money into it. I expect a lot more. So,
are there any questions from the audience? We do have a question. There you go.
Just what is the present patent situation for liraglutide?
Liraglutide expires, I think I had it on a slide, 2022, 2023, and then you have some production patents, so you have a tail of expirations.
So in practice, when can you launch without running into problems?
That will not be a problem as far as we see it, and also I should point out that we will Novo uses it in bacteria production, and they have a lot of patents regarding that. That's the problem for the biosimilar manufacturers. So the biosimilars are making it in synthetically, and we will use a synthetically produced liraglutide. It's very important to point that out, and that opens also the doors for us to, if they have already the biosimilar manufacturers taking that investment into a process for making synthetic liraglutide, I mean the additional investment to make something together with us, it's not that great. So that's a good sales argument when we are talking to potential partners for this.
Well, Marcus, thank you very much. The
Thank you.
time's up. You can, of course, be available also to answer questions after the presentation.
Yes, I will. Yeah.
Okay. Thank you very much.
Thank you.
And moving on. There has been quite a few interesting projects within drug delivery and so on. So, but now off to something completely else. I'm pleased to say hi to Edvard Hall, CEO of Bioextrax.
Yes.
Welcome.
Thank you. Yes, as mentioned, my name is Edvard Hall, and I'm the CEO of Bioextrax. We do something slightly different than most companies that are involved with the BioStock. We do industrial biotechnology. And industrial biotechnology means that you use microbes in different processes to produce different kinds of materials. And in our case, we use bacteria for selective hydrolysis of protein-rich materials. So that means that we use bacteria to break down materials in a selective manner. And when you do that, that means that you can upcycle that material. But what I want to do here today is, first of all, to present which kind of materials we can produce with our technology. We can produce a pretty wide range of different materials. We can produce a bio-based and biodegradable plastic called PHA, or polyhydroxyalkanoates. We can produce fibers. We can produce feed ingredients or protein-rich feed ingredients. And we have a number of patents applications in different regions of the world. The business model that we're pursuing is to license our technologies to different material producers. We're not planning to own or operate any facilities ourselves. And the reason why somebody would want to license from us is that we combine cost effectiveness with environmental friendliness. And it's this combination that is the basis for our business model and business idea. And since we were spun off from the Department of Biotechnology at Lund University approximately 6 years ago, we have gradually transformed from an R&D base to a commercially based or commercially focused company. And it is to accelerate this transition that we're now doing in IPO to bring in 5 million Swedish krona. But I would like to spend some more time on what we do. As a company, and as I indicated, we can produce a number of different materials. We have a number of different business areas, but they're based on the same underlying concept. So we call it a platform technology. And one of the things that we can do is that we can take any kind of organic material. So this is any material with a lot of carbon in it. This could be molasses from the sugar industry. It can be crude glycerol, which is a byproduct from biodiesel production. It can be municipal organic waste. It can be food processing wastewater and a number of other kinds of materials. And through one single process, we call it the PHA process, we produce two different materials. It's bioplastic, PHA, and a co-product from that, which is a hydrolyzed protein, which can be used as feed. Another raw material that we work with is feathers. These are feathers from chicken or turkey. And we can turn them into microfibers, keratin-rich microfibers, and also into feed. And then there are a number of other applications, such as extracting oils from algae or hydrolyzing insects and turn them into a highly digestible feed that we've done in the lab, but we haven't started the commercialization of yet. So it is these applications that we're currently focusing on, the valorization of feathers, the two different processes from that, and the production of PHAs. And I'm going to start with PHA. And PHAs are interesting for two reasons. First of all, they are bio-based and biodegradable. So from an environmental perspective, they are excellent. They also have characteristics which make them suitable replacements for up to 70% of all fossil-based plastics. And this combination, of course, is very interesting, both from a sustainability and a commercial perspective. And this is how PHAs are produced. You start with any kind of organic material, as mentioned, and here I've used molasses, which is a co-product from the sugar industry. And based on this carbon source, you grow bacteria. And after a certain amount of time, you cut the access to nitrogen. And then the bacteria can't grow anymore. And what they instead start to do is that they start accumulating the carbon in the surroundings in the form of polymers intercellularly. This is not conceptually, at least not very unsimilar to how we humans store fat as an energy reserve for the future. So what you have after this step is bacteria filled with plastics. And we are not only one who can do this process, there are a number of other companies who can do it as well, but where they struggle is how to get the plastics out of the bacteria, because of course you need to do that in order to use it for something useful. And what they do is they do a combination of solvents or chemicals, heat and pressure to disrupt the cell walls and to release the PHA. What we instead do is that we add another bacteria. We can call it the Bioextrax bacteria. And what the Bioextrax bacteria does is that when it comes into contact with the PHA-producing bacteria, it releases a cocktail of enzymes that breaks down the cell walls of the PHA-producing bacteria. And out of this, we get two products. We get the PHA, the bioplastic, which is released, and we get the co-product in the form of a hydrolyzed single-cell protein. This is the broken down cell walls of the PHA-producing bacteria. And it's important because only around, or never more than 90%, but often between 50 and 70% of the total output from the process is PHA, which means that the rest is the co-product, and you want to use it for something useful. And as it's made up of 50 to 60% of protein, which means that it's good for feed, you don't want to ruin it by using chemicals in the extraction. So our competitive advantage compared to other methods for producing PHA is mainly the cost, and this is mainly thanks to the co-product that we produce, but we also have lower amounts of energy, or we use lower amounts of energy and no chemicals, so it's cheaper from that perspective. It's also more environmentally friendly, again, because we use very little energy and no chemicals. And then we also produce PHA granules or plastics with a higher molecular weight. And that's important for certain applications. You open up new application areas, and it also makes the processing of the polymers easier. But what does the market for PHA look like? Well, currently it's still fairly small. Last year, around 550 million Swedish krona worth of PHA was sold in the world, according to Markets and Markets. But it's not PHA or the PHA market as it is, but the potential in the PHA market that is interesting, because it's generally considered the bioplastic with the highest commercial potential, amongst others by the European Bioplastic Association, and there are leading industry experts that mean that if we can cut the cost for producing PHA, we can increase the market by 40 times within a few years. And this market demand is primarily driven by new legislation. One such example is the EU's upcoming ban against added microplastics in a long range of products. And what's interesting from our perspective with that legislation is that it explicitly exempts PHA from the ban. And we saw a very strong reaction from a large number of large companies when this proposal was published, and we were contacted by companies looking for a solution. And this is why this graph produced by the Nova Institute in Germany is interesting. This is the total amount of plastic produced in the world every year during the last 70 years. We're producing around 350 million tons, and that equals a market of several thousand billions Swedish krona. That's a very large number. And we do, of course, understand that we can't replace all this plastic with PHA over the coming few years, but when we can get the production cost of PHAs down to a level that is similar to that of fossil-based plastics, which we believe that we can do, then something really big will happen very quickly. And this potential within PHA is something that we see in our everyday work at Bioextrax. We're working with companies along the value chain. We're working with companies who has the who have the organic materials that we use for the production of PHA, but we also work with buyers of both products we get out of the process. So when it comes to the owners of the organic materials, we're currently doing pre-studies with 4 different companies, these carbon sources as we call them, and we're generally working with pretty big companies. The smallest we work with here has a yearly revenue of 15 billion Swedish krona. And we're also doing tests with 3 PHA current PHA producers, who are evaluating the possibility to use our methods instead of their current methods. When it comes to buyers of PHAs, we're also working with large companies. We're doing or planning trials together with companies who each are amongst the 3 biggest in the world in their respective market. And those markets are packaging, cosmetics, chemicals, FMCG, which is fast-moving consumer goods, and fashion as well. And then we're doing analysis of the co-product together with 1 European and 1 American feed producer. And on the patent side, we have received a notice of allowance for our patent in the US, and we expect a similar result within the EU soon. And that's everything I had to say about bioplastics. Now we move over to feathers. And that might seem like a small niche to work within, but we need to remember that every year 5 million tons of feathers are produced across the globe. And This is mostly being thrown away. It's considered a problem by the slaughterhouses. They pay to get rid of it. Instead, we can turn it into two useful products, and that is feed and microfibers. The microfibers are interesting because they're extremely small. They are between 30 and 200 micrometers long, and micrometer, that's a millionth meter, and they're between 2 and 6 micrometers wide. And the small size means that it's easy to mix these fibers into various materials such as polymers or plastics, for instance. They're also hollow, and the hollowness means that they have low density. So if you mix the fibers with, again, a polymer, you will significantly reduce the density of that material. And then they're also very strong. I don't know if you try to break a turkey feather, but that's pretty tough. They are flexible and strong. And those properties they take with them into the polymer you mix them with. So we're trying them in different applications, such as plastic reinforcements, in textiles, and as insulation. When it comes to the feed ingredient we can produce from feathers, it's pretty simple. Feathers are made up of 90% protein. We break this protein down to their amino acids, which means that we can feed them easily to animals because the digestibility becomes very high. And when it comes to the commercialization of feathers, we come pretty far. When it comes to the feed application, we have entered into an exclusive term sheet with one of Europe's largest waste management companies. They have more than 75,000 employees, and a full agreement based on this term sheet is being prepared. And what it means, to keep it very brief, is that they will do the production and we will get part of the profit. And when it comes to the microfibers, we haven't come that far yet. That's partly because it's a pretty new technology, but it's also because this is completely new and unique material. There are no real benchmarks anywhere in the world, and that means that we really need to start from scratch and try different applications. One of these trials we're currently doing that I want to highlight is that we're working with one of the world's largest furniture manufacturer, and they have agreed to pay for trials, and what they are interested in is this density lowering capacity of the fibers. So what will happen in the future is, first of all, that we will finalize the installation of the pilot facility that we're building in Lund. This pilot facility will allow us to produce larger samples for our customers to try, and through this we will be able to accelerate those collaborations into full commercialization agreements. And during next year, we hope that the facility to produce feed from feathers will be under construction, and we also want to enter into commercial agreements within both PHA, the bioplastic application, and within fibers. And in order to do this, we're bringing in money. We're doing an IPO to bring in 5 million Swedish kronor. We have commitments, so teckningsåtagande in Swedish, equal to 50% of the rights issue, and people who are either employed or on the board of the company will do or will represent around 50% of that commitment. And for every 2 shares you subscribe for in the IPO, you get 1 share option. And the pre-money valuation that we're going to the market with is 24.5 million Swedish krona. Thanks a lot.
Well, thank you, Edvard. Very interesting, an interesting field. I mean, talk about being in the center of development here. First of all, an IPO, 5 million Swedish kronor sounds like a small sum. But then again, we're used to looking at companies developing drugs, which is very expensive. So where will this money take you?
We're taking in 5 million in as part of the IPO, and then through if the options are used, we hope that we'll get another 3 million, and that will be within approximately a year. And this will last us through this year and next, so until the end of 2021. And after that, we plan to be cash flow positive. And the reason why we have this very low burn rate is that we're pursuing a licensing model. So we're not constructing any facilities of our own. We don't need to apply for any permissions by ourselves and so on. So we work very closely with very large companies, and they make the investments for us, so to speak. So it's personal cost or employee cost. That's the main cost we have.
And looking at the different areas that you are active within, all of them seem to have huge potential, of course. In which area would you say that you have found the biggest commercial interest from external players?
Within PHA, definitely this bioplastic application, that's where we're working with the largest number of potential customers, and that's also where we kind of receive the most attention. And that's very much in the it's upcoming area or very trendy area, if you so will. That being said, feathers are a really big problem for the industry. They don't really know what to do with it. Both the slaughterhouses and kind of the few companies that are currently trying to do something with feathers. So we kind of, from that perspective, we do address a very, very specific, although pretty small, pain in the market when it comes to feathers. So, and we are closer to the market when it comes to the feather technology, which I hope was obvious from the presentation.
Just out of curiosity, what happens with all the feathers today? Is it landfill or?
Landfill is a very common usage or usage. Some is being burned, but the melting temperature of feathers is so high that the energy recovery you get becomes very low.
Okay. And then you mentioned a big commercial interest in PHA, and looking at the production of plastics, 350 million tons, you said annually. Is that correct? And what's the prognosis for the transition into PHA, if you can say that?
It is very cost dependent. So that is the important thing here. So currently PHAs are around twice as expensive as what we will call the average fossil-based plastic. So this is what we are targeting and addressing. With the current price levels of PHAs, we're talking a growth rate of around 11%. That's not my prediction, that's the general prediction. And that's driven by legislation. But the really big things will happen when the cost comes down.
And I should also say, questions, audience, we have one. You go.
Just to sort out a confusion with me, these bioplastics PHA, they are produced from organic material. But if they are then converted into plastics, it's still the same old plastics as if they were made from petroleum. I mean, they are not as plastics. They are not more or less biodegradable.
Thank you that's a good thing to sort out. So they are highly biodegradable. So if you throw a bag made out of PHA in the ocean, for instance, it will dissolve within 6 months without leaving any microplastics or chemicals in the ocean.
But there's a lot of plastics that you really want to be static, if I may put. I mean, if I make a chair out of plastics, I don't want it to crumble within 5 years.
No, that's of course true, and that's, I mean, first of all, you can't replace all plastics with PHA. That's, there are applications where you want it to last forever and ever. That's number one. But second, if you make a chair out of PHA, for instance, it won't start to degrade in your living room. That's not how it. You need bacteria, for instance, quite a lot of bacteria, and more bacteria than what you have in your living room. Same thing if you want to produce a bottle, for instance. So if you want a soda bottle, you can make it out of PHA. If you let it stand for a very, very long time in a moist and dirty environment, it will start to biodegrade, but it takes a long time. But the main application, you're right, is where you want the biodegradability. So within agriculture, for instance, or within medicine as well, because it's biocompatible, so it can degrade in your body without causing any harm.
you said you were spun out from the university in Lund. What are the future vision for BioExtrax? It's going to be another one of those innovative small companies that get picked up by some giant in the US, or are you going to try to stay locally?
I don't. I think, yeah. Well, you know, you can always discuss whether or not a company is a good acquisition target or not. I think our company as a whole is not, because we do a lot of different things. So there is not one single industrial player who would look at us and say, yeah, they fit perfectly in what we do, because then they will get other technologies that they kind of not that they are not interested. But I think that the patents could probably be bought, but that's not what we're aiming for. We want to grow ourselves. That's the plan for the time being, at least.
Good ambition. Final question before we have to stop. The IPO, what's the timeline there?
Yeah, we plan to do it soon, so hopefully the subscription period will start by the end of February at some point, and then last for around 2 weeks after that, and then first trading day, second half of March, and it's at Spotlight Stock Market where we'll go.
So anyone interested should come and find you in the break.
Yes, that's correct.
Thank you very much, Edvard.
Thank you.
That, my friends, mean that we are going to today's last presenter. And I will pick that up here. And it should be said then to those of you who are choosing to leave now that all these presentations will also be available online after the event. And directly after the event as a one big long video file, and later we'll publish them again with all the different separate presentations published. With that said, David Westberg, CEO of Nanexa, welcome. The stage is yours.
Okay, thanks. So, well, hello everybody. I'm David, and I'm the CEO of Nanexa and have been the CEO since beginning of 2015. I've previously worked at some different companies that you probably know of: Pharmacy Upjohn, Pfizer, and Orexo, which is a medium-sized Swedish pharmaceutical company. So what does Nanexa do then? Well, we are in the sort of drug delivery market. We have our own drug delivery technology called PharmaShell. And why it's called PharmaShell is because we do create very, very thin shells on top of pharmaceutical particles or particles of pharmaceutical substances. And we focus then on making injectables that could last for a very long time and release the API, the active substance, for a certain amount of time. That could be a week or a month or even longer, actually. And what we're trying to do is to build a company based on this PharmaShell technology and link that together with common regular pharmaceutical development and make a company, a drug delivery company, based on that into the parenteral area where we do build long-acting injectables. And long-acting injectables has the advantage of being has a better convenience for the patient. You don't need to take shots every week or every day or so. You can have it one for a month. It increases the compliance of the treatment. That is actually so that many patients, even though they have very severe illnesses, they don't comply with taking the pills every day and so on and so forth. And so having a depot formulation could really improve that quite a bit. And that could itself then make the treatment even better effect. It has a better effect than the normal route of administration. And it could also decrease the side effects of the treatment. The vision of our company we feel that we're in a stage now where we can see that we can grow to a large or medium-sized pharmaceutical company or pharmaceutical drug delivery company, I should say, that is based on this PharmaShell technology, where we do focus on parenteral administered drugs. And we do that by combining the development into both own product projects that we run, and partner-driven product projects that we also get involved in and sell the license to these companies to develop their own products. And to be able to do that, we will build a structure that will be able to take the technology into clinical studies and further on into commercial manufacturing and so on. So, and I will say we're sort of in the middle of this picture, going from the smaller size into this vision going forward. And our business model is actually based on two different steps, or two different lines, I should say. First of all, we sell license to use the technology as such. And that could be then sold to different companies for different active substances, where the partner company actually pays for the development from start and on. And we will get the revenue from licensing revenue from signing fees and milestone payments. We will get paid for the work we perform in those projects, and we will later on, when the product is on the market, we will get royalty payments on those products. The other line is the product license, and that's where we by our own develop a specific product based on this PharmaShell technology, take that into proof of concept in clinical studies, and then out-license it to a partner company. That line will of course we will get much bigger revenue from such a project. It's a little bit bigger risk, but it is worthwhile, I'll say, to go that route. And in this case, we will take the cost for the development up to clinical proof of concept, and the partner company will take the project cost from that stage onwards throughout the final clinical study and the regulatory process until it gets ready for marketing. Also there we will get signing fees, we will get milestones fees, and we will get royalty on sold goods. Another way of demonstrating this or showing this is this picture, where you can see that in the first line we'll get quite a bit more milestone payments. They're a bit lower than when going for the product projects, but we can get earlier revenue from that line. And actually we have already started that with the collaboration projects that we have ongoing since last year. We get some payments during 2019, and that will increase during this year. The milestone payments from the product projects will come later, but will be bigger. And then the royalty-based income or revenue that will come when the products are out on the market. But the base for everything is patents, actually. A company like ours wouldn't be anything without patents. So we have a good patent portfolio. We received patent grants in the US actually the first day of last year. And that specific patent application, we also got a broader application granted later on last year in the US. And we also got it granted in Japan in the middle of the year about. And we have, of course, this application in other countries as well, and the process is ongoing there. We also build on continuously with more patent applications as we invent, so to say. Well, the PharmaShell, then what is it? Yes, it's a shell that I said that we build right on top of API particles, active pharmaceutical ingredient particles. And the shell is extremely thin. It's like 10 to 30 nanometers in thickness. And that's built with a process called atomic layer deposition. It's a process that we have borrowed from the electronics industry, where it's used quite a bit when making, screens, telephones, everything like it. And and it has been shown to be very, very good to use in the pharmaceutical industry. It's a process that can be run pretty much in room temperatures, a little bit elevated, but not high temperatures. It's a gas phase process. It doesn't involve any solvents at all. So it's a completely dry process where we don't really need to have any purification steps after the process, after the shell is made. So it's a very simple process and has a good potential to be quite cheap to use as well. What we then can offer, like I said, parenteral depots that we can control those or steer the release of API we have shown from about 1 week to several months actually. It has a very broad applicability. We can work with either very high soluble drugs. That could be a problem for many other drug delivery systems. We can also work with low soluble drugs. We can work with small molecules, and we can also work with biologics like peptides and proteins. And that is very important because the peptide and protein in business is something that's growing very, very steadily in the pharmaceutical business. And we can have control of initial release. That's the third bullet here. That's very important. Think about if you inject something that should stay in the body and be released over time for maybe 1 month or 3 months. It's very important that it doesn't release initially maybe 30% of what you have injected. So you really need to keep that at control of the initial release. And we have a very, very good way of controlling that. High drug load is something that's also important, specifically if you work with longer depots. It's important to keep the volume injected as low as possible. And we have a possibility here to work with about 80% drug load in our particles, and that's very, very high. Also, we have an advantage that we actually protect the actual drug substance when we have administered into the body. So it is protected from enzymes and so on. So it stays stable throughout the time it is in the depot. This is a slide showing a little bit of what I've said. This is results from animal studies. Rat studies where we have injected these particles subcutaneously, a suspension of these particles, and we have in the graph to the left, you can see that it's a 12-week study where we have injected 3 different doses of pharmacell-coated particles, and we have compared that. The lowest dose we have compared with uncoated particles. And you can see the red line that drops into the x-axis just within a few days. That should be compared with the lowest, I think the green line that lasts for about 10 weeks. And the only difference between those two is actually the shell material. It's a shell that is about 10 nanometers in thickness that makes such a difference. And that's quite neat, I think. Then going up 10 times in the dose or another 10 times, you can see that even after 12 weeks, there's still remains quite a bit of API in the system. And that suggests that we actually have depots of maybe half a year or a year-long depots. The picture on the graph on the middle shows what I said about the initial release. We have a possibility to control the initial release in a very good way. And you see the 2 lowest ones suggest that we have about 2 to 6% in initial release the first 48 hours. And that is considered extremely good values. So that's really neat. And to the right, you can also see a 1-week study that we did. It was with a different shell material and a different shell thickness and the way we processed it. So we got a 1-week depot in this case, and that is something that's very important. to be able to show that we can also make shorter depots. And that's something that we will utilize in our own product project, Nex 18, that I will talk a little bit about. Well, what has this then led to? Well, this is the project portfolio that we have right now, January 2020. In the middle of 2018, the only thing that we had was a collaboration with AstraZeneca, one of their projects. Now we have 2 projects with AstraZeneca. We have 5 other companies that we collaborate with, where they are doing so-called feasibility studies with our concept. All of them have actually been prolonged. They have gone into a deeper collaboration together with us. And many of them, as you see, are in the animal study phase. And some other ones are very, very close to entering into the animal study phase. We also, in the bottom, I just put to mark here that we also have discussions with other companies to go into these kind of feasibility studies. Now I have, hopefully 1 or 2 of these will continue into product development projects within soon. And we are targeting our own project, the Nex 18, to go into clinical study end of this year. A little bit about our product that we run with own money, so to say. It's improvement of a product that's already on the market called Vidasa, and the Vidasa is based on azacitidine, which is the active substance there. It's a leukemia indication, MDS. And today the patients are treated with 7 different injections 7 days in a row, and then they have a resting period for 21 days, and then they start a 7-day treatment again, 21 days rest, and then 7 days treatment. And that is, of course, cumbersome for the patients to go into the hospital 7 days in a row, actually. Every 4th week, they need to go to the hospital. And those injections, as you see, they are, if you see the red peaks, they get the subcutaneous injection of azacitidine that goes directly into the bloodstream and peaks very fast, and then leaves the blood also very fast. By that treatment, they also get some side effects out of the treatment, and they don't really have the substance in the bloodstream all the time. What we will do then is to exchange those 7 injections with just 1, and the substance will be released on a steady level over the 7 days. And then the next month, the patients will come into the hospital just once, get a shot of or injection of Nex 18, and so they will exchange the 7 with 1. And that is something that we've discussed with some of the clinicians that treat these patients groups, and they said this will be a really good product to take to the market, and it will ease up the situation for the patients quite a bit, actually. The project timeline for this project is about like this. We will do some preclinical studies this year, and we will enter into the clinical phase by end of this year. We will then continue into the phase 2 program, and after that have a product license by sometime during 2022. And then the partner will take on the project. But important to say, we will get milestone payments from that time and on. To be able to realize all these projects, we have an organization in line that is, I say, we cover all the different aspects of pharmaceutical product development in this very small but very knowledgeable organization. Value creating activities like this will continue with partner projects. We'll do our Nex 18. We will actually start another product project this year. We'll develop the technology as such and start upscaling. The upscaling is quite exciting also. We have some collaboration with a really large company making equipments that suits us very well. And we will, of course, continue with our business development. And this is the last slide. We right now have a rights issue ongoing. The subscription time will end 25th of February. And we'll target 45 million Swedish crowns about. And we have folders and so on outside here that you can read more about this emission of rights. And with this, thank you for listening.
Thank you very much, David. You had quite a few partnerships lined up.
Right. Yeah, I'm very excited about that because I would say last year was really a year when we really gained some interest from the industry and started all these projects. So that's really good.
And you show the timelines and so on, but seeing that you mentioned something about 1 or 2 that you're hoping for, that 1 or 2 will continue and so on. Is that a conservative hope?
You should realize that every feasibility study will not reach into product development. And since we're running, it's actually 10 different projects with these 6 partners. And I say if like two continue into product development, I think that's a good track record. If there are more, we will be fine with that as well, of course. But I wouldn't promise anything more than that.
And as far as the projects, the collaboration or the evaluation projects starting to pay off in some way in milestones or upfront payment, when is the soonest that can happen? Would you say?
Well, I'd say the soonest. I think during this year, if we enter into product development projects, milestone payments will start this year.
And like you mentioned in the last slide, you're actually going on financing round. And 40.
It's 45, yes.
45 million, okay. And how far will that get you then?
Well, the 45 will take us through the development of and the first clinical study of Next 18. We'll start the Next 20 project. We will of course continue with our partner projects, but those we will get paid for. So those are sort of neutral when it comes to economy.
They don't add on to your burn rate.
They don't add on to our burn rate. That's how it. And we should also we're actually looking at increasing our facilities, and also the organization will bring in some more competence actually into the organization.
And looking at your slide, there was something about GMP facilities.
Yes.
You're building your own.
Right. Thank you. Yes. Yeah, we're building our own GMP facility, and that's something that is critical for taking the projects into the clinical phase. We need to be able to manufacture these pharma-shell products somewhere, and since that's a unique process, we need to build it ourselves. And then, luckily enough, we are located in an area in Uppsala, the former Pharmacia headquarters and R&D area, where we actually have a good facility where this can be fitted. And so we're adjusting that facility, and now I've started to build our own quality assurance system and so on and so forth, so to be able to manufacture these in full GMP for this phase.
So we are actually inching towards the end of our time here. I should ask if there are any questions. We have a question.
Good.
Here you go.
Apparently the technology works neatly, as you say. I'm just wondering, you have very many small particles with this shell around it, and something in the tissue dissolves the shell, but only some of these particles, a few of them, and then gradually it dissolves more of those particles. How come that many particles are sheltered for a while
That is a question I also get from many of the pharmaceutical companies when I present this. Think of it as you know, even if I say that the shell thickness is has a certain thickness, there is of course a spread in that thickness. So some of the particles maybe have 5 nanometers in thickness and some have 15, and the mean is 10. Then. So that's one thing that controls that some particles release their API prior to other particles. Then there is another mechanism actually. When you inject, like you said, very, very many of these particles in one injection, and then you inject it, and there is a small lump of those particles. The particles that are on the outside of that lump has a sort of a higher tendency to dissolve faster than the ones that are in the middle of that lump. So there are two mechanisms that works together to get a nice and easy release of the API. But thanks for that question. That was really clarifying. Thanks.
Thanks for a clarifying answer.
Okay.
We are going to have to say thank you to you.
Okay,
I know
thanks.
you have some material with you that you can feel free to pick up outside. Thank you very much for coming today.
Well, thank you.
Yeah. So that will actually mark the end of our today's BioStock live session from Stockholm. Next time we're here would be in March. So tune back in then. Thank you very much for listening today.